Hi everyone. Good afternoon. Good afternoon, guys. Welcome back to Unacademy NEET English channel. My name is Tina, and we are here with very important session, guys. That is NCERT Important Line C from class 11. So we are going to complete our class 11 NCERT today in this session. So we are starting now. I think it will go up till 4 or 5. So just stay tuned, tighten your belts. It is going to be a long session, guys, but we have to read our NCERT. I have already underlined all the important lines so that we don't have to hunt for them. And these lines are based on the important questions that we have already gotten in our previous year questions, or they are expected in next exam. So good afternoon once again. Quickly give me a thumbs up that you're all set with your NCERTs. If you have already highlighted your NCERT, no worries. Don't highlight over the highlight. Just make sure that you do keep some symbols, hashtag things which I make you underline today. Is that clear? So yes, guys, please stay confident that we have learned certain things. They are already in our memory. They will be loyal to us and come back whenever we need them. But we need to revise certain things. Is that clear? So this is NCERT reading session. So quickly get your NCERTs ready. I'm going to use the PDFs here. Yes, guys. Good afternoon. Good afternoon, all of you. Quickly share the session as well with all your friends. All right. Shall we start? Will there be breaks? Yes, there will be some breaks. There will be 5 or 10-minute breaks in between, water breaks or short breaks, right? Yeah. But don't expect lot of breaks. We'll take breaks after completing some units. Okay. So let's start with the first unit, guys. This is the diversity, and we are going to start with a very basic chapter, the first chapter, that is living world. Usually get one or no question out of it, but again 2026 saw one question from the topic of binomial nomenclature. Let's go through the chapter quickly. Let's go through the chapter. Yes. All right, let me take my pen. Where is it? Just a second, guys. Just a second. Yeah. Here it is. Shall we start? Shall we start? All right. So see, on the very first page, guys, on the very first page, we see this. The number of species that are known and described range between 1.7 to 1.8 million. So, okay. Now, where do we make mistake? We mark it as billion sometimes. So they must have given 1.7 million also. They'll also give 1.7 billion. So what do you have to remember? It is in millions. 1.7 to 1.8 millions. This is the number of species that are known and described. Now moving on to next page. See here, coming here, page number 2. Here there is a need to standardize the name of living organisms, and this process is known as nomenclature. Naming is only possible when organism is described correctly. So now you should know that nomenclature is going to be the last step, right? It is described correctly, and what we know, what organism and the name is attached. This is identification. Is that clear? This is called identification. Now there is another term, ICBN. This is International Code for Botanical Nomenclature and International Code of Zoological Nomenclature. I'm only underlining, I have already underlined very important things from this chapter because we need time for all right. So now please go away. I hate this part. All right, guys. Okay. So yes, so this is ICBN. Then we have binomial nomenclature. Who gave this? This is Carlos Linnaeus. And what does binomial nomenclature say? That according to this, guys, every name is going to have 2 parts. So generally in Latin, and that is why it is italicized. So it is a common question even in assertion and reason. Why do we italicize the scientific name? Just to mention its or highlight its Latinized origin. It is in Latin. So the first and then second point you must know, are all the scientific names in Latin only? No, not only in Latin. If they put the word only after Latin, then the statement goes wrong, because some have been derived from Greek also. Now first word in the biological name represents a genus, second one is a specific. When handwritten, it is separately underlined, and when written in and when printed, it is in italicized. It is in italics. Why? To indicate the Latin origin. Just told you here. Right? Now, moving on to the next. Genus starts with the capital letter and the name of species will start with the small letter. There is this example given in here. Mangifera indica Linn. So, see, first is the generic name, then the specific name. You see it has been italicized, and the name of author appears after the specific name, Linn, and that too in the abbreviated form. Is that clear? Next. So what is classification? It is a process by which anything is grouped into convenient categories based on some easily observable characters, right? And now, all these categories, the scientific term for these categories is taxa. Categories can be at different levels. Now, this process of classification is known as taxonomy. Is that clear? See, earlier taxonomy was based on only external features, merely observable characters, but later on external and internal structure of cell, development process, ecological information, it formed the basis of modern taxonomic studies. Are we able to underline all these things? Are we focusing on all these lines? So, the classic taxonomical studies considered only the easily observable characters, that means external morphology. But modern taxonomic studies will take into consideration all these things: external structure, internal structure, structure of cell, development process, even the ecological information. Now, very important line. See, what are the processes involved in taxonomy and in what order? So, characterization, identification, classification, and nomenclature. What do you have to remember? CICN. Now, bear it in your mind always. What is it? CICN. CICN. Just remember that. It is characterization, then identification, then classification, and then nomenclature. Is it clear? These are the basics to taxonomy. Now, earliest classification, as I told you, was based on the uses of various organisms. So, earlier, people used to classify, okay, whether this thing can be used for shelter or for clothing or for eating purpose, for food, right? So the earliest classification was based on use of the organism. Then came systematics. This is also proposed by Linnaeus only, the term as well as the branch. Systematic word comes from the Latin word systema, which means systematic arrangement of organisms. Who is the father of taxonomy or systematics or binomial nomenclature? It is Linnaeus, right? And he described this in his book Systema Naturae, right? Takes into consideration evolutionary relationship between the organisms also. Now look at these taxonomic categories. All the categories together constitute the taxonomical hierarchy, and each category is the unit of classification, and each category is known as taxon, right? Then category basically denotes the rank of the organism. Now, taxonomic groups or categories are the distinct biological entities and not merely morphological aggregates. So, actually they exist. Is that clear? Are we, if in case you have missed on any line, please make sure. And if you have underlined the whole paragraph, please remember that you know those lines and make sure you highlight these things or just put a hashtag over it. Is that clear? Now, species as a species is the lowest taxon or the lowest category. Now, what is species? Group of individual organisms with fundamental similarities. Now, according to the present proper definition of species, it is a group of organisms who can interbreed, right? Now, see, morphological differences will be present, distinct, which will differentiate them from the other species. For example, some are here. So species are indica, right? Tuberosum. In Panthera leo, it is the leo part. So all these indica, tuberosum, leo represent the species. While Panthera has another species also, like tigris, like Solanum has species like nigrum. Is that clear? Like Melogena. Now, human beings belong to the genus is Homo and our species is Sapiens, right? So, we write it as Homo sapiens. Genus, group of related species. Potato and brinjal, different species but same genus, that is Solanum. Leo, Pardus is leopard, and Tigris is tiger. But they have a common genus, that is Panthera. And then cats, the genus of cat is Felis, right? Now family is formed by the group of related genera. You must know these examples. So the name of family will always be indicated by the suffix AC. So there is the name Solanaceae, Poaceae, right? We have seen so many names, Leguminosae. So how do you indicate the name of the family? It is by this suffix ACEAE. This is what indicates it. Then Felis belongs to the family Felidae, dog family Canidae. These are the things you must remember. Now, order, similar characters are less as in number as compared to different genera. So you have to remember, as we go up the taxonomic categories, the characteristics of common characteristics, they go on decreasing from species to kingdom. So kingdom will have least number of common characteristics, right? So Convolvulaceae, Solanaceae, Convolvulaceae and Solanaceae, they belong to the order Polymoniales. You have to remember that. Similarly, your Felidae and Canidae, these are the two families. This Felidae is the cat family, Canidae is the dog family. They belong to the order Carnivora. Then class, order class is, see, our class is class Mammalia, right? But the class will have many orders. For example, order Carnivora of dog and cats, and our order is Primata, right? In Primata, we include monkey, gorilla and gibbons. Then in phylum, guys, in phylum, coming here, so phylum, we have phylum Chordata, right? But in case of plants, we don't use this term phylum, we use the analogy. What is that same category named as? Division. For plants, we have division. For animals, we have phylum. And then coming on to kingdom, see, as we go higher, told you, as we go higher from species to kingdom, the number of common characteristics go on decreasing. Lower the taxa, more are the characteristics that the members will share. Higher the category, greater is the difficulty of determining the relationship to the other taxon. Is that clear? So this is the figure of taxonomic categories in the which order? Ascending order. So this is the smallest and this is the highest. I hope this one is clear. Now coming on to this table, NCERT loves this table. Okay, NTA loves this table. See here. So man, Homo sapiens, Homo hominidae, Primata, Mammalia and Chordata. So recurrently we have got questions on housefly. The name is Musca domestica and the family is Muscidae. Order is Diptera, Insecta and Arthropoda. Mango, Mangifera indica, Mangifera anacardiaceae, Sapindales, Dicotyledone and Angiospermae. Triticum aestivum, Triticum poaceae, also known as Gramineae, Poales, Monocotyledone and Angiospermae. Are we clear till here? Please let me know this point is, this chapter is clear. So guys, yes, we have quickly revised the first chapter. Yes, please tell me. The first chapter is done. Now moving on to the second chapter, that is biological classification. So here we have just read in the chapter of living world that earlier classification was based on usefulness of the organism. Okay, now see, then Aristotle gave the earliest scientific basis for classification. So the first and foremost scientific classification was given by Aristotle. See, used simple morphological characters and divided plants into trees, shrubs and herbs. Divided animals on the basis of whether they have RBCs or they don't have RBCs. What was the basis of his classification? Simple morphological characters, right? Then came Linnaeus and he gave two kingdom classification, Plantae and kingdom Animalia. This system, see the disadvantages or the criticism, did not distinguish between eukaryotes and prokaryotes, unicellular, multicellular, photosynthetic and non-photosynthetic. Is that clear? Then besides the gross morphology, other characters used were cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationship. These were also included in later on classifications. Is that clear? But Linnaeus did not involve these. The only criteria used by Linnaeus was the presence or the absence of cell wall, and we will see it here. Yes, is this point clear to all? Now coming on to this very important table, characteristics, 5 kingdoms. So which of these kingdoms, out of the 5 kingdoms, which kingdom belongs to Monera? Prokaryotes, that is only Monera, and rest all the kingdoms belong to eukaryotes. See the cell wall composition. In Monera, it is non-cellulosic, made up of polysaccharides and amino acid. Let me know in the chat what is the name given to this. We call this polysaccharide plus amino acid as peptidoglycan, right? Then Protista may or may not have cell wall, and that too made up of different components. Fungus has cell wall made up of chitin, while plants have made of cellulose, and animals do not have cell wall. Nuclear membrane will be present in all except in Monera. Body organization, see here, cellular, multicellular, tissue and tissue organization. In the paper of 2025, guys, we have seen this question in which they have asked us to arrange the kingdoms in a particular sequence from lower to higher or better body organization, right? So you must know the body organization very clearly. They have given certain characteristics of each kingdom and asked us to arrange them in the correct sequence. So learn these properties very carefully. Cellular, fungus has multicellular or loose tissue, plantae has tissue or organs, while Animalia will have tissue, organ or organ system. Is that clear? While mode of mode of nutrition, autotrophic, and we have all types of nutrition here. Autotrophic, also heterotrophic. In auto we have chemo and photo. In hetero also we will have saprotrophic, parasitic and even symbiotic. So autotrophic as well as heterotrophic. Fungus is only heterotrophic. Plants are always autotrophic. Animals are always heterotrophic. Are these points clear? Now this is R.H. Whitaker in 1969 giving us 5 kingdom. What were the main criteria for his classification? See the criteria here. Cell type, cell wall, nuclear membrane, body organization and mode of nutrition. Is this point clear? Now see, it's written here as well. Cell structure, body organization, mode of nutrition. Other than that, what is written here? Next is reproduction and phylogenetic relationship. They have not included this. Is this clear? So reproduction and phylogenetic relationships are also included. Then 3-domain system came up, in which that divides the kingdom Monera into 2 domains, right? And only 1 domain was dedicated to eukaryotes. So 2 domains given to prokaryotes. How? See, and thereby from this 3-domain system we got 6 kingdom classification also. Is that clear? So which kingdom was further subdivided? It was kingdom Monera, and it was divided into arc bacteria and eubacteria. Arc bacteria domain 1, eubacteria domain 2, and rest all the eukaryotes were put into domain 3. Are these points clear to everyone? Please tell me. Are we underlining these lines? Right? So guys, I have already completed the detailed marathons of all these chapters. Today I am just going to read NCERT and highlight the important lines. If there is any doubt, please make good use of chat and ask relevant questions. Is that clear? Now, then earlier classification included, see, according to Linnaeus classification, they are saying earlier classification, bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, angiosperms, all were kept under plants. So it also grouped the unicellular organisms and the multicellular ones together. For example, Chlamydomonas and Spirogyra were placed together under algae, right? So is this clear? Now, there was no difference between autotrophs as well as heterotrophs. Even there was no difference on the basis of cell wall. Even though fungus has cell wall made up of chitin that we have read, and green plants have a cellulosic cell wall. Now moving here on this very important line, kingdom Protista has brought together Chlamydomonas and Chlorella, which was earlier placed in algae with plants. But later on it was pulled out of the kingdom Plantae, and Chlamydomonas and Chlorella has been categorized under Protista, right? And now both have cell walls. And Paramecium and Amoeba, which were earlier placed in animal kingdom and lack cell wall, they were taken out from kingdom Animalia and put under Protista, protozoans that we call them today. Is this point clear? Is this point clear? Right? Now, moving on to the kingdoms individually. So guys, these are the diagrams. By the way, I will be taking a separate session on all the diagrams where we will be analyzing each and every diagram and seeing the possibility of questions from each and every diagram. Is it clear? That session is also aligned in the coming days. So please stay tuned and do not miss any session now. Is that clear to all? Right? Okay. Now, moving on to the next to see this cocci, bacilli, spirilla and vibrio. So here are certain lines. So bacteria are the sole members of kingdom Monera. Extreme habitats like hot springs, deserts, snows, deep oceans, they can be found here, divided into 4 categories on the basis of shape. So you must know the basis of classification always. Is this clear? Now see, these are the very important lines. These are very important lines. It is written, bacterial structure very simple, complex in behavior and most extensive metabolic diversity. Please underline these lines. So it is very simple in structure, right? Very complex in behavior and most extensive metabolic diversity, which are all these 3 statements are true for bacteria. Is that clear? Now, talking about arc bacteria here. So arc bacteria found in the extreme conditions. For example, extreme salty areas, halophiles, hot springs, thermoacidophils, marshy areas, methanogens. Why are they able to survive these extreme conditions? Because of different cell wall structure, right? They are responsible for their survival. Then methanogens, where are they found? In the gut of several ruminant animals like cows and buffaloes. For methane production, they are found in the biogas. Is that clear? And these are the main places where they are found. In the biogas plants, in the gut of ruminant animals. Are these points clear? The third one I've already told you, it is also found in the marshes. Clear? Now, talking about eubacteria, see, they have rigid cell wall and they may or may not have flagella. If they have flagella, they will be motile. Now, cyanobacteria, referred to as a blue-green algae. It has chlorophyll A similar to that of green plants. What is the structure like? Unicellular, colonial, filamentous. Now, cyanobacteria is a photosynthetic autotroph. Is that clear? It is surrounded by, see, the mucilaginous or the gelatinous sheath, right? Often form blooms in the polluted water bodies. Very important line. Some of these can fix atmospheric nitrogen, and these specialized cells where nitrogenous fixation can occur is known as the heterocyst. Now, examples of 2 heterocyst containing BGA, that is nostoc and enamina. So they both look like this. They are colonial, they are filamentous. Are these points clear? Noted? Very good. Now, chemosynthetic autotrophic bacteria, these ones, they will derive their food. How will they are autotrophic? So how do they make their food? They oxidize various inorganic substances like nitrites, nitrates and ammonia and use their energy for ATP production. And second function of chemosynthetics are recycling of nitrogen, phosphorus, iron and sulfur. Very, very important lines. Heterotrophic bacteria, the most abundant out of phototrophic, chemotrophic, heterotrophic is the most, and the heterotrophic one, sorry. So heterotrophs are the most abundant bacteria, they and majority are decomposers. So out of autotrophs and heterotrophs are more, and in amongst heterotrophs, decomposers form the majority. Is that clear? They also help in making curd, antibiotic production, fixing nitrogen. Is that clear? For example, rhizobium in legumes. Is this clear? Some are pathogenic, parasitic, so they'll cause disease, cholera, typhoid, tetanus, citrus canker and diseases. So from diseases, I remember guys, tomorrow also I'll see you at this time, 12 to 4 probably, and we are going to read class 12 NCERT. So make sure that you hit the bell icon so that you get a proper notification. And today's special class has been rescheduled for 9 p.m. tonight. Otherwise our time is 2 p.m. but because of today's session I have scheduled it for 9 p.m. tonight. Is that clear? So we have already completed NCERT Exemplar class 11 in our special classes. Today I'm going to start with class 12. So let's have a quick reading now. So moving on, so bacteria reproduces mainly by, which is the most convenient and the most common method for division, that is fission. Under unfavorable conditions, it will produce spores. Favorable conditions, it will prefer fission. In unfavorable, it will produce spores. Now, the sexual reproduction is very primitive type, and it involves transfer of DNA from one bacteria to another. This is actually conjugation that they are talking about. Now there is this unique type of bacteria which is called mycoplasma, the smallest living organism, lacks cell wall, smallest living cell, survive without oxygen, pathogenic in nature. You have to remember these 4 points about mycoplasma. And because they don't have cell wall, they can keep changing their body shape. Pleomorphic. We have read that in our marathons already. Are these points clear to all? Yes. Now, kingdom Protista. So all single-celled eukaryotes. What are the type of organisms involved included in Protista? Single-celled, but not prokaryotes. They are eukaryotes. So who are eukaryotes? They have membrane-bound nucleus, and they have other membrane-bound organelles as well. Is it clear? So, but the boundaries of this kingdom are not well defined, because how was Protista created? You must know. So later on, organisms from Plantae and kingdom Animalia were pulled out to form this. So there are no well-defined boundaries. Now this kingdom forms a link with the other, dealing with plants, animals and fungi. Are these points clear to all? Please let me know. Are these points clear to all? Right? Now, Protista reproduces asexually as well as sexually, and will involve a process called cell fusion and zygote formation. Now, starting with the first type, which is chrysophytes. There are two types of chrysophytes. One is diatom, second is golden algae or desmids. They will be found in fresh water as well as in marine. They are they float passively. That means they cannot choose their own direction. They will move according to the mercy of water, according to the mercy of tides. So hence float passively, and such organisms are known as planktons. Most of them are photosynthetic, and they are very important. Two thin overlapping shells, and the shells contain silica, and these two overlapping shells will fit like soap box. Very important term. You will not hear about soap box anywhere else in whole biology. The walls are indestructible because of silica. Now that is why the earth, which contains remnants of diatoms, is known as diatomaceous earth or kieselguhr. What are the functions of diatomaceous earth? Used for polishing, filtration, for filtration of oils and syrups, also for making soundproof elements. Is it clear? And because they are photosynthetic, hence they are the chief producers in the ocean. Are we clear till here? Yes. Now moving on to the next type, which is dinoflagellates. Wait. Yes, please. So why do we call them dinoflagellates? Because they will have two flagella. And what type of? We'll just see. So mostly marine and photosynthetic. Main pigments present in their cells, which are yellow, green, brown, blue, red. They have stiff cellulosic plates. In diatoms, the shell contains silica, but now the cell wall contains cellulose, right? They have two flagella, hence the name dinoflagellate. But one flagella, see, is longitudinal, while the other is transverse, flowing through. So this is longitudinal one, and the other goes through this transverse groove between the cell plates, and this one is the transverse one. Now, very often red dinoflagellates, for example, goniolux, it causes red tides. Rapid multiplication is shown, causes red tide because of the pigment. But these red tides are very toxic. Goniolux produces toxins which can kill other marine animals. Are these points clear? Please let me know. Are these points clear to all? Now, the third type of Protista that I'm going to discuss is euglenoid. We are going to talk about euglenoid. So this is euglenoid drawn here. So it is fresh water. Most of them are, majority are found in the fresh water. Now, not running water. It is always found in the stagnant water. Now, it does not have a cell wall. Instead of cell wall, it has a protein covering known as pellicle. Now, like dinoflagellates, these also have two flagella, but one is extremely short and the other is actually long. Short and the long one. Photosynthetic in the presence of sunlight and becomes heterotrophic when deprived of oxygen. Sorry, deprived of sunlight. So, in the presence of sunlight, it is an autotroph. In the absence of sunlight, it is an heterotroph. That is why we use the term mixotrophic. We don't use the term mixotrophic for this, and this is one character which makes it a connecting link between animals and plants. Is that clear? Now, pigments of euglenoids are identical to those of higher plants. Please tell me all these points are clear to you. Right? Now, moving on to the next type, which is slime mold. This is the slime mold drawn here. Saprophytic. They are all saprophytic, right? Now, so move their body along the decaying twigs and leaves engulfing the organic material. Now see, under suitable conditions, their aggregation is known as plasmodium. So when they are in the suitable conditions, favorable conditions, they will aggregate to form plasmodium and it will spread a lot, which grows to several feet. But in unfavorable conditions, this is the plasmodium, and plasmodium is multinucleated. Is that clear? And there is no cell wall here. There is no cell wall here in the plasmodium. But in unfavorable conditions, it will form some fruiting bodies, and these fruiting bodies will bear spores at their tips, and the spores will have true walls. They will be extremely resistant, and under adverse conditions, right, they can survive for years. See, spore or otherwise also, spores are very resistant structures, right? And the spores are dispersed by wind or air currents. Please tell me that these points are clear to you all. Is that clear? Right? Now, moving on to the next type of protista, which is protozoans. All the protozoans are heterotrophs. They are predators and parasites called the primitive relatives of animals, right? Now, they are further divided into 4 categories. Now, the 4 categories are on the basis of locomotory structures, right? On the basis of locomotory structures. The first one being amoeboid. So they move and capture their prey by putting out pseudopodia or false feet, just like in amoeba. Now, marine forms will have silica shells on their surface. For example, ant amoeba is one such form which is parasitic. Is that clear? Amoeba is free living. Marine ones will have silica, and the ant amoeba is a parasitic form. Is that clear to all? Now, moving on to next, which is okay, why is this not going? This is not going anywhere. Oh guys, what have I done? All right, now, okay, let's not disturb it. Okay, so flagellated ones are those which will have flagella for their locomotion. They may be free living or parasitic. The example given here is of parasitic type, trypanosoma, which causes sleeping sickness. Then the ciliated one, it will move actively because of the presence of cilia on their outer surface, and the cilia helps in 2 things. It not only helps in the locomotion, it also causes the water laden to bring food into its gullet, and the example is paramecium. And then we have the 4th type of protozoan, which does not have any specific locomotory structure, guys, and that is a sporozoan. It is a sporozoan. Now see, sporozoan is a spore-like stage and the most notorious form. Why do we call it a sporozoan? There is a spore-like stage in its life cycle. For example, in Plasmodium, we know there is a sporozoite stage, right? Which is a malarial parasite, causes malaria. Are we clear till here? And next, coming on to the kingdom fungi. Let's read more about kingdom fungi. See, constitutes a kingdom of heterotrophic organisms. You must remember, you have seen moist bread and rotten fruits because of fungi. Now see, the most common mushroom you eat and the toadstools are fungi. Toadstools are not edible, mushroom is edible. Then parasitic ones, white spots on mustard are due to a parasitic fungus. What is this fungus? This is Yes, guys, this is albugo they are talking about. Then yeast makes beer and bread. We know that fungi causes diseases also. For example, rust causing puccinia, and there is other Eustilago also, which causes smut. Now, antibiotic producing fungi is penicillium and others as well. Then they grow best in the warm and humid climate. With the exception of yeast. Yeast is unicellular, otherwise they are multicellular and filamentous. The long slender thread-like structures known as hypha and their network known as mycelium. This hypha can be cenocytic if there will be no cross walls. Others which have cross walls will be uninucleated. So they are called septate, and these ones are always aseptate. Is that clear? The cell wall will have chitin, and chitin is itself a polysaccharide. Is that clear? Now, they are heterotrophic. They absorb soluble organic matter from the dead substrate. So, they are saprophytes, which depend on living plants and animals, are parasites, which live nicely in association with other organisms. They are called symbionts. For example, with algae, it forms lichens, and with roots of higher plants, it forms mycorrhiza. Are we clear? Reproduction takes place by vegetative means, fragmentation, fission, and budding, and asexual reproduction by spores. Now, the asexual spores are, so these are the examples of asexual spores. So, the asexual spores are conidia, sporangiospores, zoospores. Is that clear? So, we have conidia, sporangiospores, zoospores, and also aplanospores in case they are non-motile. Yes, and for sexual reproduction we have, or sexual spores are boa, basidiospores, oospores, and ascospores. So I hope you are writing down these tricks to learn. Please let me know. Are you writing down all these points with me? And at the end of the session, guys, you will or keep a copy separately, right? Don't keep on writing those tricks and keep telling me that in the end of the session, how many tricks have you got today? Is that clear? Let's do that. Now, so these are the sexual spores that I've told you already. Boa, it is oospores, ascospores, and basidiospores. Now the various, Now the various spores are formed in the distinct structures called the fruiting bodies. So the body in which spores are formed are called the fruiting bodies. Now, sexual cycle involves the 3 steps. The first step is plasmogamy, second is karyogamy, and third is zygotic meiosis. Now, we have seen question around this topic multiple times. You can be asked, right, which of the following is the correct sequence. So, what is the correct sequence of these steps? First is plasmogamy, followed by karyogamy, and then followed by zygotic meiosis. See, plasmogamy gives rise to dikaryon phase, N plus N. And when these two N will fuse, it is known as karyogamy. And when N fuse, that is karyogamy occurs, it results in the formation of 2N structure called zygote. And then 2N structure called zygote will undergo meiosis. So zygotic meiosis. And meiosis will give rise to the formation of, gives rise to 4 spores. Is that clear? But in ascomycetes, guys, it will later form 8 spores. Now, so these 3 steps, this special type of sexual reproduction in which plasmogamy and karyogamy does not occur simultaneously. This occurs in only 2 classes of fungi, which are ascomycetes and basidiomycetes. That is why dikaryotic stage is formed in these only. It is called a dikaryon. Now, very important. See, this is already a PYQ, guys. This is already a PYQ. So, the morphology of mycelium, mode of spore formation, and the fruiting body, they form the basis of division or classification for the kingdom into various classes. So there is this question, which of the following was not a basis, is not a basis for classification? Mode of nutrition is not a basis. That is why I have written here, because it is a PYQ, and the question was exactly this: which of the following is not a basis of classification? So mode of nutrition is not a basis of classification for kingdom fungi. Is that clear? Now see here. So first of all, let's talk about phycomycetes. So phycomycetes found in aquatic habitat, decaying wood in moist and damp places, and obligate parasites. If you have attended all the marathons, guys, you know that I have given you a proper table of these four classes, and we have compared their habitat and all other things, right? Is that clear? Please let me know, are these points clear? Dropper, when will in dropper batch NEET 27 will start? Srija, it will start after Re-NEET exam. But the classes have already started. If you have taken a batch, then you can start attending the classes. Okay? But a dedicated batch like dropper batch will start after Re-NEET. Is it clear? Okay. Now see. So aquatic habitat, dead, decaying, moist and damp places, they are obligate parasites on plants. Very important line. And the mycelium is aseptate and cenocytic. It is only Phycomycetes which will have aseptate and cenocytic condition. Rest all other classes will be septate and branched. Now see, zoospores, motile, aplanospores, non-motile, they represent the asexual spores, and these are always formed endogenously in a fruiting body called sporangiospore, or sporangium, sorry, not sporangium, called sporangium. Zygospore is their sexual spore formed by the fusion of two gametes. Now, Phycomycetes represent three types of gamete condition. It can be isogamy, it can be anisogamy, or it can be oogamy also. Now see the examples. I've given you the mnemonic already. Yes, how many of you remember that mnemonic? How many of you remember that mnemonic? So for Phycomycetes, we have PRAM. Now you know what PRAM stands for. So write down separately. So PRAM, Phycomycetes, it is rhizopus, it is A for albugo and M for mucor. Rhizopus is the bread mold and albugo is the parasitic fungus on mustard. Are these points clear to all? Then guys, for Ascomycetes, we have stamp CN. Is it clear? Now, see here. All right. So for Ascomycetes, we have stamp CN. So this is this mnemonic has been made by one of my students in our paid batch, Preeti. So she made this awesome one. Otherwise, I never found a mnemonic for Ascomycetes. Anyways, so Ascomycetes, commonly known as sac fungi, mostly multicellular. But there will be penicillium, which is the unicellular one. Sorry, the yeast, which is the unicellular one. Penicillium is multicellular, by the way. So saprophytic decomposes coprophilous. Now this term will be present only in Ascomycetes. It is coprophilous, means growing on dung. Now branched and septate and sexual spores are conidia, and conidia are always exogenously formed. Sexual spores, ascospores. Ascospores are endogenously formed in a sac called esi. Fruiting body, ascocarp. See, the spore is ascospore, the sac is esi or ascus, and the fruiting body is ascocarp. Carp word is always related to a fruit, fruiting body. Is that clear? So some examples are Aspergillus, Claviceps, Neurospora. Neurospora used in biochemical and genetic work. This is a PYQ in match the following. Morels and truffles are the edible ones called and considered as delicacies. Then we have Basidiomycetes. For Basidiomycetes, we have a mnemonic called Bapu, right? Now see, commonly known as mushrooms, bracket fungi, puffballs found in soil, on logs and tree stumps. Asexual spores generally not found, and the sexual spores are basidiospores. Basidiospores are exogenously formed on basidium. The fruiting body is known as basidiocarp. The examples: A for agaricus, P for coccinia, and U for eustilago. Is that clear? Then coming on to Deuteromycetes, we remember it as DCAT. DCAT. This one is known as imperfecti fungi because only asexual stages are present, sexual are not present. And later on, when perfect stage is the term given for sexual stage. Perfect or sexual stages, when were found, were moved to Ascomycetes and Basidiomycetes. So later on they were discovered, but they were so dissimilar from the parent body, from Deuteromycetes, their characteristics matched more to Asco and Basidiomycetes. Is that clear? Now, The asexual reproduction is the only type of reproduction here, and it will occur through conidia. Is that clear? Fragmentation though will occur, but I'm talking about asexual and sexual. So sexual, either not present, if found, move to other classes. Is that clear? Now, so asexual reproduction occurs through conidia, and conidia are always formed exogenously. Mycelium is septate and branched. They can be saprophytes or parasites. Very important, decomposers of litter and helps in mineral cycling. And the examples are CAT, that is Coleotrichum, Alternaria, and Trichoderma. Please tell me these examples are clear to you all. Yes, guys. And then we have kingdom Plantae. So members are partially heterotrophic. Some members, not all. Few members partially heterotrophic. Otherwise, they are all eukaryotic chlorophyll containing organisms, and hence autotrophic. While bladderwort, so some examples of insectivorous are bladderwort and Venus flytrap or pitcher plant, right? Then Cuscuta is an example of parasitic plant, amarbale or dodder. It does not have, it is not autotrophic. So it directly withdraws nourishment from the host plant. Is this clear? Now the life cycle of plants will have two distinct phases, be it any type of plant, algae, bryophyte, pteridophyte, we'll talk about it, but they will have two distinct phases. The diploid one is known as sporophytic phase, and the haploid is known as the gametophytic phase, and they will always alternate with each other, no matter what. One can dominate over the other, one can be predominant, but they will always alternate with each other. Is that clear? Now this is known as alteration of generation. Are these points clear to all? Are these points clear? Now kingdom Animalia. Yes. So kingdom Animalia characterized by all heterotrophic eukaryotes. They're all multicellars and they will not have cell wall. The store form of food is glycogen or fat. Remember this. They have holozoic mode of nutrition. That is, they take in the whole food, digestion occurs within the body, and the waste is removed through egestion. And they have a definite growth pattern because we don't have any meristem, right? And higher forms will have elaborate sensory and motor mechanisms. Now please tell me these points are clear. Now moving on to these acellular organisms. Acellular because they'll behave, the body is not made up of cells, but they'll behave as organisms when inside a living host, which are virus, viroids, and prions. Now in 5 kingdom classification, there is no mention of lichens and all of these acellular organisms. Virus is non-cellular, inert crystalline structure outside the living cell. This is a non-living feature. So these are the structures given in here. Now see. All right. So see, viruses, what does virus means? It means venom or poisonous fluid. Dmitry Ivansky in 1892 discovered viruses. The causal organism of mosaic disease of tobacco, TMV, was the first virus discovered. They are smaller than bacteria and passes through bacteria-proof filters. Is that clear? Then Bajer-Naik named them. So who gave the term virus? It was Bajer-Naik, named the new pathogen virus and also called them contagium vivum fluidum, infectious living fluid. Stanley could be crystallized. So I showed that viruses could be crystallized or crystals consist largely of proteins. They are inert outside the living host cell. Viruses are obligate parasites. Are these points clear? Then no virus contains both RNA and DNA. Virus is a nucleoprotein structure. The genetic material is infectious. Plants have ssRNA. Animals can have single or double-stranded RNA or double-stranded DNA. And bacteriophages will usually have double-stranded DNA. Then the protein coat is known as capsid, which is made up of smaller subunits like capsomeres. These capsomeres can be arranged in helical or polyhedral manner, and because they are parasites, they will cause disease like mumps, smallpox, herpes, influenza, AIDS. In plants, guys, the symptoms will include mosaic formation, leaf rolling, curling, and yellowing of leaves. And yellowing and vein clearing. Is that clear? Then dwarfing and stunted growth. Then in 1971, Theo Dinier discovered viroids. They are different from virus. Smaller than virus, cause potato spindle tuber disease free RNA. So there is no protein code. It lacks protein code that is found in virus. So because there is no protein code, also the low molecular weight. Are these points clear? Then coming on to prions, known for neurological diseases. Now viroids don't have protein code, and prions, they have proteins only and no genetic material, but these proteins are abnormally folded proteins. The size is similar to virus and they cause disease like bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and the analogous variant is Creutzfeldt-Jakob disease, CJD, in humans. Is it clear? Talking about lichens, these are symbiotic association between algae and fungi. Algal partner known as phycobiont, fungal partner known as mycobiont. Is that clear? They are very good pollution indicators and do not grow in the polluted areas. Please tell me this point is clear, this chapter is clear. You have underlined all. Yes? So we are done with the second chapter. All right. Moving on, guys. Moving on to the third chapter now. Third chapter, that is plantae. Let's talk about plantae. All right. Let's talk about plantae. Yeah. So coming here. Yes. So see here, that influence that the earliest classification of systems of classification was based only on gross superficial morphological characters like sorry, yes please, like habit, color, number and shape of leaves, right? And they were based on vegetative characters only, for example, or the androecium structure, not vegetative but sorry, who's thinking of me? Anyways, mainly on vegetative structures or the androecium structure. The only reproductive structure which was considered here was androecium structure, and this was given by Linnaeus. Now this system was known as artificial system. Sorry guys. Yes please. Is that clear? All right. So artificial system given by Linnaeus based on gross superficial morphological characters like habit, color, shape of leaves, mainly that is the vegetative characters, or on the androecium structure. Is that clear? This is artificial. A for artificial, A for androecium structure, right? Now, the artificial system gave equal weightage to vegetative and sexual characters. Now, this is not acceptable. Why? Because vegetative structures are more easily influenced by the environment. So, we got natural system of classification given by Bentham and Hooker. So, Bentham and Hooker not only considered the sexual characters, also took into consideration internal features like ultrastructure, anatomy, embryology, and phytochemistry as well. Right, then we got phylogenetic classification given by Engler and Prandtl based on evolutionary relationships between the various organisms. Numerical taxonomy carried out using computers based on all the observable characteristics. So hundreds of characters can be considered. Numbers and codes are assigned to all characters, and then data can be processed using the computer. So it gives, in this way, each character is given equal importance, and at the same time, hundreds of characters can be considered. In cytotaxonomy, cytological information, chromosome number, structure, behavior are used. In chemotaxonomy, the chemical constituents of the plants are used to resolve the confusion, that is for classification. Are these clear? Now, starting with the first type, that is algae. So, algae and the most primitive plants, right? Chlorophyll bearing, simple, thalloid. Thalloid means body not differentiated into true root, stem and leaf. Autotrophic and largely aquatic, fresh or marine. Moist stones, soils and wood. This is a habitat. Can be just free living or found in association with fungi, that is in case of lichens, and even with animals. So this is a symbiotic association given here. So symbiotic association, two examples of algae, one in association with fungi forming lichen and second with animals, for example, sloth bear. Is that clear? Now, see the form. So, highly variable form can be colonial, for example, volvox, filamentous like Eulothrix and Spirogyra, and some are very massive, for example, kelps, hundreds of meters height. Then, vegetative reproduction by fragmentation, asexual by zoospores, or if non-motile, there will be aplanospores, right? Then sexual reproduction through gametes. So in gametes, there we will see three types of condition. If both the gametes are similar, we call it isogamy. If both are dissimilar, we call it anisogamy. And if female gamete is large and non-motile and the male is motile and small, this is called oogamy. Now see the examples here, guys. See the examples. So gametes can be flagellated and similar. Flagellated and similar. That means they are talking about flagellated and flagellated isogamous. So this is isogamy and flagellated condition. This is seen in case of Eulothrix. Is that clear? Then isogamy and non-flagellated. So this is isogamy and non-flagellated, that is Spirogyra. Is that clear? Then an isogamous example is Eudorina, and for oogamous we have volvox and fucus. Are these points clear? Are these points clear? Right? Let's move on now to the next. So these are the diagrams that you must be able to identify. This is volvox, that is Eulothrix, this is laminaria, this is fucus, and that is dictyota, right? And this one is porphyra, this is polysiphonia. We'll talk about diagrams in more detail in a separate session. Now, algae, very important paragraph, very important paragraph. See, increase the level of DO, dissolved oxygen, in the immediate environment, number one. Then porphyra, laminaria and sargassum, they are among the 70 species of marine algae which are used as food. Then hydrocolloids, water holding substances, algin and brown algae. Carrageen gives us red algae. This is already a PYQ. How do you remember? So we all remember RRR. See here, we have RRR, RR, RR. So algin is from brown algae, carrageen is from red algae. Can we remember it like this? Agar comes from Gelidium and Gracilaria, both are red algae. Chlorella, unicellular algae, used as a food supplement for space travelers. And now algae can be divided into three categories based on their pigments basically. Now see, so chlorophyll, chlorophyce has chlorophyll A and B. Chloroplast can be discoid, plate-like, reticulate, cup-shaped, spiral and ribbon-shaped, and they have some storage bodies known as pyrenoids. Very important. What does pyrenoid contain? Pyrenoid contains P for protein, and then starch is usually present in the plants. So remember starch. So pyrenoid is made up of protein and starch, right? Now, the stored form of food can be oil droplets also. Is that clear? In some cases. Then green algae has a rigid cell wall, inner layer of cellulose and outer layer of pectose. So you have to remember that inner layer will always be made up of cellulose, be it green, brown or red. Inner layer is always of cellulose. Outer layer will be different in all the three. For example, in chlorophyce it is pectose. We have just seen, right? Then vegetative reproduction by fragmentation. You cut the filaments and they will all regrow into a whole new plant. Asexual reproduction by zoospores, and need not to mention that zoospores are always flagellated. Zoo word is for motility, right? Then it shows iso and oogamy. See the examples: Chlamydomonas, volvox, Eulothrix, Spirogyra and Cara. Then Phaeophyce, simple branched filamentous, for example Ectocarpus, massive like kelps. See the pigment A, C. Carotenoids and xanthophylls, and among xanthophylls they have special xanthophyle, that is fucoxanthin also. We'll see it in the table also. See, xanthophyle pigment called fucoxanthin. Now the food is stored in the form of laminarian starch or mannitol. Is that clear? So mannitol is a type of starch. Mannitol, it is an alcoholic sugar. Is that clear? Now gelatinous coating of algin on the outside. See, inner coating is always of cell wall. In cell wall, the inner coating is always of cellulose. Outer coating pectose in case of chlorophyce, and in phaeophyce it is elgin. Plant body is now slightly differentiated. Not true stem, it has hold fast, it has stipe, it has frond, leaf-like structure, frond. Is that clear? And then fragmentation occurs here as well, zoospores present, but now the flagella will be pear-shaped, the flagella will be lateral, one short, one long, two unequal, laterally attached. Iso and isoogamy, see the examples here. Ectocarpus, Dictyota, Laminarian, Sargassum and Fucus. Then coming on to rhodophyce. So red pigment are marine, found in the warmer waters. Is it clear? Both well lighted as well as close to the surface of water, also at depth. So they'll be found in all the zones. Complex body organization stores food in the form of Floridian starch, similar to, now the structure of Floridian starch is similar to amylopectin and glycogen. So if you remember that biomolecules topic, so we have seen the detailed structure of glycogen, highly branched. Amylopectin is also a branched form of branched chain of starch. So means Floridian starch is highly branched, right? Fragmentation, non-motile spores, there'll be no flagella. Sexually also by non-motile gametes. Is that clear? So this table, very important, very important table. Everything that we have read so far has been tabulated in this table. Is that clear? Is that clear to all? And now sexual reproduction will be only of oogamous type. Is that clear? Oogamous type with complex post-fertilization development. Example C, P2G2. So what is the P2G2? Polysiphonia, Porphyra, Gracilaria and Gelidium. Are these points clear? Yes guys? Everyone clear? Please let me know. All clear to everyone? Yes? All right, perfect. Now moving on to bryophytes. So in bryophytes, we are going to talk about two categories, mosses and liverworts. So this is Markensia shown here, which is unisexual, so different thallus. This is the female thallus, this is the male thallus. So see the structures, rhizoids, gamma, archegonia, this is entheridiophore, this is archegoniophore, this is archegonia, this is entheridium, right? And this is the funaria shown in here with gametophytic and sporophytic body, and this is sphagnum. Okay, we'll come on to the diagrams later. Now bryophytes, also called the amphibians, right? Why? Their life cycle is totally dependent on water, especially the sexual reproduction. They'll always be found near water, right? Now very important, they play important roles in plant succession or bare rocks. Can you give me some tea? Tea. Please, tea. Tea. Please, tea. Happens. Sometimes you have to beg for certain things in life, and there is no problem in begging. All right, guys. Oogamous and how non-motile gametes, Anjana. See, this is an exception. You have to remember this. In case of red algae, the gametes will also be non-motile. It is a special case. I know you're talking about males being motile, but no, not here. Is that clear? All right. Now, let's talk about the economic importance. They play a very important role in plant succession on bare rocks and soil. So it is thallus-like, unicellular, multicellular rhizoids. They may possess root-like, leaf-like, stem-like structures. The main plant body of bryophyte is haploid. Is that clear? Now it produces, what haploid means, gametophyte. And what does gametophyte do? Produces gamete. Remember that. Haploid, GH comes together in English alphabets. So haploid is gametophyte, and gametophyte will produce gametes. Can you remember this? Yes, haploid is gametophyte and gametophyte produce gametes. So gametophyte will always have sex organs. We have done the details with all those flow charts. I hope you have revised them. Sex organs are multicellular. Male sex organ antheridium produces biflagellate entherozoid. Sex organ called archegonium is flask shaped, produces a single egg. Entherozoids are released in the water where they come in contact with archegonium. Entherozoid fuses with egg to produce a zygote. Now, why have I underlined and highlighted all these lines? Because this is a question in 2025 exam. So we got this question in 2025 PYQ. Which of the arrange the following steps in correct order of their life cycle? So you must know the events in proper sequence. Is that clear? Zygotes do not undergo reductional division immediately. They form a multicellular body called sporophyte, not free-living but attached to the gametophyte to derive nourishment. Sporophyte undergoes reductional division and it occurs in the capsule area and forms spores, and spores will germinate to form gametophyte. Now, food for herbaceous mammals, sphagnum, a moss, provides peat. So it's also known as peat moss. Basically, sphagnum can absorb a lot of water. So when it dries up, it loses its whole water. It can be used as fuel, right? And as a packaging material for transshipment. See, why can it be used as a packaging material? Because it can store water. Is that clear? Then it prevents soil erosion, and these are the ecological importance. So bryophytes are divided into liverworts and mosses. So let's first talk about liverwort. It is thelloid, or for example Markensia. Asexual reproduction in liverworts is by fragmentation or by specialized structures called gamma. Very important. Gamma, green multicellular asexual buds present in gamma cups. So if gamma is present on the male thallus, it will give rise to a male thallus, gamma. If gamma is present on a female thallus, it will give rise to a female thallus. Very simple, right? Now, during sexual reproduction, we know what will happen. All right, talking about the mosses here. Predominant stage of life cycle of moss is a gametophyte, just like that. But now the gametophyte has two further stages. First is the protonema stage, followed by the leafy stage. See, first is protonema, which is creeping, green and branched and frequently filamentous. Second is the leafy stage, which develops from the secondary protonema as a lateral bud, right? And leafy stage has spirally arranged leaves. Are these points clear? Then vegetative reproduction, again by fragmentation, then budding in the secondary protonema. Sporophyte is more elaborate than in liverworts. Very important lines. And they have elaborate mechanism of spore dispersal. Example is it is FPS, Funaria, Polytrichum and Sphagnum. Please tell me these points are clear to you all. Are we able to revise everything? Yes, Anurag? Please tell me, guys. All clear? Nostalgia is saying, you made me think my whole existence. What happened, nostalgic universe? What is so nostalgic here? The concepts of class 12. So NCERT sessions. Will it be helpful to score 340? Jyoti, see, if you are very thorough with the syllabus of class 12, it is okay, but I mean, it totally depends on you. If you have already been scoring 340 plus in your all tests, then maybe you can skip, right? But make sure that you are practicing questions every day. Make sure that you are taking the mock tests every day. You're solving one paper every day and you are able to consistently score between 340, 360. Then it is fine. Then this can help. Then work on your shortcomings only. Okay? Then we can do that. Is that clear? All right. Now coming on to pteridophytes. So pteridophytes include horsetails and ferns. Medicinal purpose So they are used for, they have medicinal purpose, they are soil binders, and also they are ornamentals. Found in cool, damp, shady places and in sandy soils. Main plant body is a sporophyte and the body is differentiated into true root, stem and leaves. Is that clear? And it has vascular tissues. No, thank you. बहुत बढ़िया. Thank you. Is that clear? All right, guys. All right. So not true stem and leaves, they are well differentiated vascular tissues, and they are small microphylls known as Legionella. If the leaves are small, we call them microphylls. For example, in Legionella, and the large leaves known as macrophylls will be found in ferns. Now, some of them all of them, they have the special leaves, spore-bearing leaves called sporophylls. In some of them, the spore-bearing leaves, that is sporophylls, will form a compact structure called strobili or cone. Is that clear? But not in all. In some cases, sporophylls can form strobili cones, for example, Legionella and equisetum. Now, spores are formed within sporangia, where meiosis occurs in the spore mother cells, right? Now, spores are small and spores will give rise to inconspicuous small but multicellular. See, this is the strobili of equisetum, right? Okay, we'll come back to these diagrams later. It gives rise to free-living, mostly photosynthetic thalloid gametophyte called prothallus. Gametophytes bear male, female sex organs called antheridia and archegonia. Water is required for the transfer of antherozoid. Again, a PYQ. Arrange the following events of the life cycle in correct sequence, guys. So, for solving these type of questions, you should actually refer to those flow charts of marathons that we have discussed and learn them in correct order. Of course, it is a life cycle, so events will happen in a proper sequence. So, you have to learn them in a proper sequence. Is that clear? Okay. Then, guys, gametophytes bear male, female sex organs called antheridia and archegonia. Water is required for the transfer of antherozoids. Fusion of male female with the egg present in archegonia result in the formation of zygote. Spores of similar kinds, homosporous. So algae, bryophytes and most of the pteridophytes, they are homosporous, right? But few exceptions will start occurring here. For example, Legionella and Salvinia. See here, Legionella, Salvinia are heterosporous. Very important line, female gametophytes in these plants are retained on the parent sporophyte for variable period. Now this will form a base for seed habit also. Is that clear? So development of zygote into young embryo take place within the female gametophyte. This is a precursor to the seed habit as an important step in evolution. Very important lines. So they have further divided into 4 classes, guys. See, Psilopsida, Lycopsida, Sphenopsida and Pteropsida. So now do you see the sound here, opsida, right? So opsida word, this suffix is used to classify or name the classes, right? So one we have seen AC during the living world chapter. AC, Solanaceae, Leguminosae, Poaceae, right? Fabaceae. So AC is for family, Opsida is for classes. Is that clear? Now, you must know these examples and they have already been asked in PYQ. Now, talking about gymnosperms here. Gymno means, so this 2026 paper on 3rd May which happened, we got just one question from plant kingdom and that question was this, around gymnosperms. So why are they naked? Because they are not enclosed by any ovary wall. So seeds that are post-fertilization, not covered and are naked. Sequa is one of the tallest and generally, now these gymnosperms generally have taproots. Some will form symbiotic association like Pinus. In case of Pinus, it will form mycorrhiza. In case of Cycas, there will be coralloid roots. There is symbiotic association with nitrogen fixing cyanobacteria. Is it clear? Stems can be unbranched like in Cycas and branched in case of Pinus and Cedrus. Leaves can be simple or they can be compound. Leaves are well adapted to withstand the extreme conditions, right, of temperature, humidity and wind. needle-like leaves, thick cuticles, sunken stomata. These are all their adaptations. Is that clear? To reduce the water loss. Now, gymnosperms and angiosperms will be completely heterosporous. Then they will form a lax or a compact strobili or cone, right? Now, microsporophylls, male spore-bearing leaves, and um, megasporophylls, female spore-bearing leaves. They will form cone-like structures, male cone or female cone. So they will form male strobili. Male gametophyte generation is highly reduced and confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. Very important line. By the way, this line is also true for angiosperms. Male and female cones may be on the same tree, like Pinus, hence Pinus is bisexual, while Cycas will have different plants. Male plant different, female plant will be different. So Cycas is unisexual. Is that clear? Megaspore mother cell is differentiated from one of the cells of nucellus. This is very similar to that in case of angiosperms, right? It is, nucellus is protected by envelopes and the composite structure is known as ovule. Megaspore mother cell divides meiotically to form the spores. And again, 4 cells will be formed, 3 will degenerate, the same story. That bears 2 or more archegonia or female sex organs. We are talking about the ovule. Is that clear? Pollen grain is released from the microsporangium, carried in by air current. So only wind pollination occurs here. In contact with the opening of the ovules born on the megasporophyll. Now, pollen tube carrying the male gamete grows towards the archegonia and discharges their content near the mouth of archegonia. Now, zygote develops into an embryo and the ovules into the seeds. Seeds are not covered. Seeds are not covered. Is that clear? Now, let's talk about angiosperm here. Okay. Yeah. See here. So, angiosperms. Pollen grains or ovules are developed in a specialized structure called flower. Now, we don't have cones in angiosperms, we have flowers, right? The sporophylls, the condensed form of sporophyll will be known as a flower here. So, flower will give rise to a fruit. So, seeds are enclosed in the fruits. Smallest angiosperm is Wolffia, the tallest one is Eucalyptus. So, they provide food, fodder, fuel, medicines and several other important, and 2 classes, dicotyledons and monocotyledons. Is that clear? So, as with this, we complete the chapter of Plantae. Please tell me that this topic is clear to all of you. Let's have a quick tea break. Yes, guys, all those of you who are reading the chapter with me, let's take a minute and message me in the chat. Yes, guys. Wow, my husband makes really good tea. All clear? Yes? Time for the green hearts then? Good afternoon. Hi, Mahesh. Finally, you said you'll join me at 1. You're joining me at 1:22. Anyways, so we have done 3 chapters. Please watch the first 3 chapters later on in the recorded form. And now we're going to start with Animal Kingdom. Is there any doubt in the previous lectures, guys? And let me know in the chat, how is your preparation going on and what are the challenges you are facing right at this stage as well? Can you do anatomy now? Hithal, why? Any particular issue? Like, yes, Haripriya, plant cycles and all those things, they are in your syllabus, and that is why I have discussed all those things in my marathons. I've discussed them already. I'm using the new NCERT here, but I have discussed all the topics in detail, right? Perfect. Plant cycle and alteration of generation is there. Oh yes, Venkatesh. I'm very lucky to have him. I'm very lucky. It's the gift of my life. Yes, I will share this PDF. All right. Let's start with the fourth chapter, that is Kingdom Animalia, Animal Kingdom, right? Coming on to the levels of organization, all members of Animalia are multicellular, yes? In sponges, loose cell aggregate is present and they exhibit cellular level of organization. And then later on there will be tissue level and organ level. Organ level is exhibited by platyhelminthes and others. And then annelids, arthropods, mollusca onwards, it will be organ systems. So functional systems will be formed, right? Now coming on to open system, guys. Sorry, open type. Blood is pumped out of heart, cells and tissues are directly bathing in blood. In closed, there is a series of vessels of different diameters. So basically, in open and closed, the basic difference is around capillaries. Here capillaries are present, here capillaries are not present. Arteries and veins can still be present here along with, or as in closed. Is that clear? But capillaries are only found in the closed system. Now talking about the symmetry. Guys, give me a second. Yes, guys, there is some issue here. I am here, but there is some issue. There's been a short circuit here. My screen is gone off. Good afternoon, those of you who have joined me now. So there is some issue with the screen, guys. Let's wait for a minute. Yes, till that time we can talk. You can let me know about your challenges that you were telling me. Yes, challenges that you are facing. And I just realized what I need to do. Yes, guys. Are we all here? Yes, please. Ma'am, what is your favorite tea brand? My favorite tea brand. I'm not really a very brand conscious person, be it anything. I just like, if I like something, I like it. And for tea brand, it has to be made by my husband. He makes good tea. That's the brand I like. It can be anything. But yes, I always like flavored tea. I can't take tea without a flavor, be it like fennel or cinnamon, anything, or elaichi. It has to be flavored. Ma'am, life cycle alteration or generation is there or deleted? Haripriya, alteration of generation is definitely in your syllabus. Just that one page given in old NCERT, that has been taken out of the new one. But how can you take out alteration of generation from the plants? Plants will show alteration of generation. So you cannot take out that alteration from the life cycle of plants. So while we are studying it in detail, we have seen that sporophytic generation is altering with the gametophytic generation, yes? So has to be there. So anything else? Okay, Mahesh. Okay, I'll see your message. By the way, Mahesh has won yesterday's practice session. So Mahesh deserves the gift. So yes, you have messaged me on Telegram. I'll revert back. Siri, how much? Venkatesh. Venkatesh is getting... Okay, guys. Everybody please contact Venkatesh. He's buying you, or I think he's gifting you MBBS seats for free, or he's charging something. I don't know. Venkatesh, if government of India finds out about this, nexus that you're running in the live chat, I don't know what's going to happen here. We are keeping the teachers, we are keeping the experts who are making papers in, house prison like conditions. They can't use phone, they can't use internet, they are in complete lockdown conditions, right? And you are running an excess here. Wow. Yeah, there will be class. Give me 5 minutes. Let me see if things go well. I'm getting it checked, guys. I'm getting it checked. If it takes more than 5 minutes, then we'll have to maybe replan something. But give me just 5 minutes. Okay? Is the new NCRT sufficient? Yes, Isha, new NCRT is absolutely sufficient. You don't have to buy the new NCRT. Not now and not even for NEET 2027. You will get everything in the class. We have discussed the topics which are not in NCRT. We have discussed them already in our marathons. And if you want, I can share that the old NCRT PDF on my Telegram channel. By the way, the Telegram channel is Tina Ma'am Biology, right? TINAMAAM Biology, right? I'll share it there also. But otherwise, you don't need them really. I am only your video of, ma'am, I am only your video of biology, Nagesh. You have to explain what you are saying. Okay, yes, Nagesh, it is sufficient. It's not beta plated, it is beta pleated. Yes, nostalgic. What are we talking about here? How much time it will take? If light comes here, I was planning to complete it by 4 or 4:30. I hope God doesn't have any other plan. Thank you, Venkatesh. Oh, yes, guys. Ma'am, your marathons are really helpful. Thank you, Amrut. Yes, guys. Power of, there is power, but there is no power in the in this in my screen. So there's been a shortcut here. Sorry, short circuit here. I'm getting it checked. Give me just few minutes. In case it doesn't get resolved in the next 5 minutes, we'll have to reschedule, but please cooperate for 5 minutes. Okay? Next test when? Tonight, 9 p.m. Okay. Then I have 20, 24, 25. Can I follow this or should I follow new NCRT? Note, are you appearing in 26 Re-NEET exam? Then why will you buy new NCRT at this point? You know, new biology, new NCRT book will be also new book for you. It won't feel like your own. You have to make your books your own, you know, by underlining them. You have to develop that bond, those markings. That is your bond with your book. You buy new NCRT, it will feel like, oh, what am I studying now? Don't buy any new book right now. Hi, Casper. Hi, all the Kerala students. We have class on 4. Yes, I know you have class at 4 p.m. I know that. And that is why I was planning to complete the session before those classes. Oh, thank you, Asman. It got written new syllabus. Yeah. Sound will be all right. Just give me some. My books have whole ecosystem in them. Oh, yeah. There should be whole ecosystem. Yes, guys. Please let me know. See, if not, if we have to reschedule this class, I really don't know. Hi, Ansika. Then we'll have to put it for tomorrow. Because in the evening, I already have my plus class also. Otherwise, I could have taken it. Or maybe we can do one thing. Maybe tomorrow, if we have to reschedule, if another 5 minutes light doesn't, if this is not sorted, then probably we'll do one thing. We'll take 2 sessions tomorrow, one 12 to 4, and then tomorrow's evening as well. We can complete so that our things are not delayed and the schedule stays as tight as. What do you suggest? Can you give me a whole day tomorrow? Haripriya, I've already answered your question, bache. Oh, Anurag is furious. Anurag and Tanvi are fighting. I am trying to continue, but the screen is not working. There's been a short circuit, that's why. Start at 10 a.m. tomorrow. Is there any way to continue the class? Kirtana, I am dying to do that as well. If I'm not able to do it now, then I won't be able to take it at 4, because at 4 p.m. you already have, I think, chemistry class planned or physics class planned. One of the classes is planned already. Mm. One of the classes is planned already. I think, what I... Laksh, I'll give my thoughts on such things post Re-NEET. Okay? First, KK sir's class is planned at 4 p.m. now, and then 7 p.m. onwards, I already have a class. 7:15, I have a plus class already. Except population attributes, rest all the topics. Oh, it's back. We won't have to reschedule. Let's restart the class. It's back. Thank God it has started working. Okay. God heard us. No, we are not doing it tomorrow. If everything stays fine. Oh yes. It's back and we are going to read it now further. Thank you so much for cooperating, guys, and staying with me. How did I solve it? Thank you. I'll do it. हाँ, बाहर भी है. Okay. Thank you. Magic? Yes. You need some magical people in your life to do some magics. Two more minutes and it'll all start again. Quick. Should I beg this board also? Okay. You're doing this chapter now. Okay, we are here. Yes, thank God. We didn't have to postpone the class. All right, guys. All right. So we were discussing about open system and we were talking about the closed system, symmetry. So sponges are mostly asymmetrical. Okay. Sponges are mostly asymmetric. Then radial symmetry is seen in coelentrates, ctenophores, and echinoderm adults, not the larva, but the adult ones. Annelida, arthropoda, and all the other, they will show bilateral symmetry. That means only in one plane the body can be divided. So diploblastic have two germ layers, ecto and endoderm. That is porifera, coelentrata, and ctenophora. And undifferentiated mesoglea is present. Mesoglea is present between ectoderm and endoderm. While in case of triploblastic, which have third germ layer called mesoderm, from platyhelminthes to chordates. Now this paragraph is very important. Coelom, right? The body cavity which is lined by mesoderm, but remember on both the sides. If mesoderm is present only on one side, it is a pseudocoelom. Annelids, mollusca, Annelida onwards, we have true coelomates, right? To chordata. Body cavity not lined by mesoderm, instead mesoderm present in scattered pouches. This is pseudocoelom, only found in Aschelminthes. Is it clear? Then animals in which body cavity absent is called acoelomate or platyhelminthes. Is that clear? See the diagrams. Then segmentation. So body, you need something? Okay. So the body shows this pattern called metameric segmentation and the phenomena is known as metamerism. Notochord is a mesodermally derived rod-like structure present on the dorsal side, right? Where's the electrician is here? Just a second. I'll get it sorted. Switch off.
नहीं पहला हाइड्रोजन बंद करना अभी बंद तो नहीं करना
Okay. Okay. Let me know. Okay. Yeah, electrician is here, so it will be completely sorted, don't worry. Now, so the classification of animals. Now let's see further this diagram, this table. So cellular level only in porifera, radial and acoelomates are coelenterata and ctenophora. Bilateral but acoelomate, platyhelminthes. Bilateral but pseudocoelomate is esculminthes. Bilateral and coelomates are rest of them. Is that clear? Now talking about porifera. Porifera, cellular level of organization, water canal or transport system, presence of ostia. The body, this body central cavity is spongocoel. It is not a body cavity, right? It is just a central cavity, spongocoel, and the water goes out through osculum. Now, very important function of water canal system: food gathering, respiratory exchange, removal of waste. Three functions given in here. Conocytes, collar cells line the spongocoel. Digestion, intracellular, spicules or spongin fibers, right? Hermaphrodite. Then reproduce asexually by fragmentation. Fertilization, internal. And where exactly? In the mesohyl layer, between outer and inner body wall. Is that clear? Then development is indirect, larval stage, morphologically distinct from the adult. You have to remember these lines. You have to remember these lines. Are we all here, guys? Now see the examples: Cycon, Spongilla and Euspongia. Now see the phylum, Coelenterata or Ctenophora. So see, nidoblast or nidocytes, stinging capsules or nematocysts. Is that clear? Anchorage, defense. What is the function of these nidocytes? They are meant for anchorage, defense and capture of prey. See, whenever a function is given in your book, right, always write it as 1, 2, 3. Never write it in lines like essays. Don't write essays. But your mind will always capture that picture in a better way if it is written 1, 2, 3. Learn them as, you should know how many functions, 1, 2, 3. Okay, so if you have not done that previously, try it now. So what are the functions? Anchorage, defense and capture. So ACD you have to remember: anchorage, capture and defense. Is that clear? Then the central gastrovascular cavity, single opening called mouth on hypostome. Digestion, extracellular as well as intracellular. Some will form corals which will have calcium carbonate in their skeleton. Polymorphism is seen. So two basic body forms, at least polyp and medusa, will be seen. Hydra and Adamsia, they represent sessile and cylindrical form, that is polyp, and umbrella-like and free swimming stage, that is Aurelia or jellyfish. So now there is an alteration between polyp can form medusa, medusa can form polyp. This is called metagenesis. Is this clear? Now, then examples are Physalia, Portuguese man of war. The common names are also important: Adamsia, sea anemone, Penatula, sea pen, Gorgonia, sea fan and Meandrina, brain coral. Now, phylum Ctenophora. Ctenophora, known as sea walnuts, exclusively marine. I have already asked you to do this exercise. If you have still not done it, guys, please do it now. This is very, very important. Make a list of all the phylums so just write down a heading right now: exclusively marine. The first phylum, put it down there: 1, 2, 3, 4, 5. There will be 4, 5 things that I'm writing there. Exclusively marine. First one is Ctenophora, right? Now diploblastic, 8 external rows of ciliated comb plates. What is the function? Locomotion. Second, bioluminescence, they emit light. Reproduction takes place only by sexual means. Fertilization, external, indirect development. Example, pleurobrachia and tenoplana. Platyhelminths, bilaterally symmetrical, dorso-ventrally flattened body. Triploblast, in fact, these are the first triploblast. Acoelomates, hooks and suckers. Flame cells, sexes are not separate. Fertilization is internal. Example, tinea and fasciola. Are these clear? Then talking about esculminthes, circular in cross section, roundworms, that is why we call them. Organ system level, pseudocoelomate, because mesoderm lines the body cavity only on one side, or you can say scattered pouches. So muscular pharynx, alimentary canal is complete for the first time, and it has developed muscular pharynx. Excretory tube removes the body waste. See, in case of platyhelminthes, we have flame cells. We have flame cells here, specialized cells, flame cells for osmoregulation. While here, they have special tubes. See, excretory tube to remove the body waste from the body cavity. Now, sexes are separate also for the first time. Till platyhelminthes are completely bisexual, hermaphrodites. Now begins the unisexuality. They'll have different sexes. Is that clear? All right. Now, males and females are distinct. That means they show sexual dimorphism. Female longer than males. Fertilization internal, and development can be direct or indirect. So guys, please go through NCERT with me now. And after the end of the whole session, I'm going to share a few tables with you where I have summarized the whole chapter of Animal Kingdom. Okay, I'm going to share that table with you, but only those who are going to attend this session will be able to get it. Okay, so yes, so you have to attend the session, you have to attend the special classes, and after special class, guys, I'm going to share that whole table. You may not even have to read NCERT after this, after that table. It is such an elaborate, good table of Animal Kingdom. Okay, so we'll do that. You'll get it after 10 p.m. So tonight 9 to 10 I'm going to join you with more practice questions, and I'm going to summarize the whole chapter in a detailed table. Good one. Now see, Ascaris, Wuchereria, and Chylostoma. Then Annelida, metamerically segmented body, segments known as metameres. The word Annelida comes from annulus, possess longitudinal circular muscles which help in locomotion. So first time these longitudinal and circular muscles have come up. They also possess some lateral appendages. Aquatic Annelida. Nereis has lateral appendages called parapodia, and because it is aquatic, the parapodia will help in swimming. Closed circulatory system for the first time. Only two phylum will have closed circulatory system. One is Annelida, second will be Chordata. Right? Nephridia for osmoregulation and excretion. Neural system consists of paired ganglia, lateral nerves, double ventral nerve cord. Nereis aquatic form is dioecious, means unisexual. Male, female are separate. Earthworm leeches are monoecious, means bisexual. They are hermaphrodite. Reproduction is sexual, and these are the examples. This is your Nereis, this is your leech. Now, coming on to phylum Arthropoda, largest phylum. Over 2/3 of all species on earth are arthropods. And we also know, when we were reading through ecology, we have read, see, out of 10 animals, 7 are insects. So out of arthropoda also, majority are the insects. So 2/3 animals are insects. Is that clear? Then it is covered by chitinous exoskeleton. Body consists of head, thorax, and abdomen. While in few of them, body will consist of two parts also, where head and thorax will combine together to form cephalothorax. Right? Like in case of your palaemon as well, that is prawn. Now, arthros means jointed appendages. Respiratory organs, gills, book gills, and book lungs, or trachea. Is that clear? Now, circulatory system, open type. Sensory organs, antennae, eyes, compound as well as simple. Statocyst. Stato word is for balance. Statocyst are balancing organs. Excretion takes place through malphigian tubules. Now, this is a line where you have to slow down and understand more. See, we have discussed it in detail that there are different types of excretory organs, but NCERT here, it is creating this, very confusing statement. It appears as if, according to NCERT, here, if we are reading this line, it appears as if all the arthropods will excrete through malphigian tubules, but we know this is not the case. There are coxal glands, there are antennary glands also, green glands. So those details have been mentioned in other chapter. So we know that, right? So here excretion takes place through malphigian tubules, but not in all. Insects will excrete through malphizian tubules, while some of them will do it through antennary glands, green glands, while spiders will have coxal glands also, right? Now, mostly dioecious, unisexual, means male, female, separate, and they also show sexual dimorphism. Fertilization usually internal, mostly oviparous, but sometimes it can be direct or indirect development as well. And sometimes it can be viviparous. By the way, scorpions, they are viviparous, they directly give birth to the young ones. Economically important, honeybee, apis, bombyx, silkworm, lacifer, lark insect, vectors, anopheles, culex, aedes, gregarious, they move in groups, locusta or locust, living fossil which has not undergone an evolution, that is limbless or king crab. Second largest is mollusca, has calcareous shell, unsegmented body, distinct head, muscular foot and visceral hump. Now the spongy layer of skin known as mantle, space between hump and mantle is mantle cavity. Now this mantle cavity contains feather-like gills. What are the function of these gills? It is respiratory as well as excretory. Noting down, then sensory tentacles on the head region and mouth has this file-like rasping organ known as radula. Please tell me these points are clear. Please tell me these points are clear. Yes, guys? All right. Now, moving on to the next part, which is usually dioecious, means unisexual, oviparous, indirect development. See the examples, very important. So, Pila, apple snail, pink tada, pearls, oysters, sepia, cuttlefish, loligo, octopus, aplysia, sea hare, dentalium, tusk shell, and Chetoplura, chiton. Is that clear? Now, recently I was just going through these questions. I'll be practicing these questions with you in the special class also. So there was this question, which of the following organisms belong to the phylum Mollusca? And all the names were starting from C. Sea hare, sea pen, sea this, sea that, right? So you should be able to, distinguish between these names. So write down, make a list of all the organisms which start with C or which end with the name fish. Please make sure that you do these things. Okay, so, and how many animals? You miss one animal and the question goes wrong. So be very careful about such questions. Now, phylum Echinodermata, endoskeleton of calcareous ossicles. So all are marine. So second phylum, exclusively marine. First, Ctenophora. Second, Echinodermata. Okay, so we got two names here. Adults radially symmetrical, larva bilaterally symmetrical. Now it has water vascular system. Porifera, it had water canal system. This one has water vascular system. And see the function, 1, 2, 3, 4. Locomotion, capture, transport of food and respiration. Is that clear? Excretory system absent, sexes are separate, sexual reproduction, fertilization usually external, development indirect with free swimming larva. See, we have talked about two larvae here. One, in case of Porifera, larva is morphologically distinct from adult. You remember that line? And here it is saying free swimming larva. Which one? Echinodermata. Is that clear to all? And then asterias, starfish, sea urchin, sea lily, cucumeria and ophura. Yes, guys. Please tell me that all these points are clear to you all. Quick. Are these points clear? Clear to all? Yes. Now, talking about hemichordata. See, hemichordata has this stomochord, rudimentary structure in the collar region. What is that? Stomochord. So many questions. So stomochord present in which region? Proboscis, collar or trunk? It is the collar region. By the way, this labeling is wrong and we have corrected that already. Okay, is that clear? So stomochord is present in the collar region and similar to notochord. And this really created a lot of fuss earlier. People thought of it as a notochord, but later on it was understood that it is not notochord. because it is first of all found on the roof of the buccal cavity, and unlike notochord, which is mesodermal, this one is endodermal. Is that clear? Now, phylum worm-like consists of worm-like organisms, and again marine. So exclusively marine. Third, Ctenophora, Echinodermata, and Hemichordata, exclusively marine. Are you noting down these things? Please let me know. I know, guys, most of you are not very serious in the chat, so I don't even bother to read this chat a lot. I just go through it once so that, you know, the serious ones and the honest ones are not missed out here. But those of you who are actually attending the session very honestly, please don't forget to leave messages on the Telegram or in the comment section later on, so that I know how many of you are getting benefited. It is indeed a big motivation. You know, your message is the biggest appreciation and the motivation for us to keep going on and bring such amazing sessions, so that, you know, this is what we earn at the end of the day. Keeping aside the finances, they are of course needed for leading a good life, but this is also important. You know, this is the real motivation. So please do leave your comments how you find the session, what all new things you want us to bring, what else you want us to bring up here, right? So please do use this as a, you know, tool to reach out. Now, then bilaterally symmetrical coelomate, proboscis, collar, and trunk. So circulatory system is of open type, open type, gills present. Proboscis gland is excretory in nature, external fertilization, indirect development, melanoglossus and secoglossus. Now, chordata, see here, see the diagram. This diagram itself is very important. This is nerve cord, and do you see there's this orange thing? This shows it is hollow in nature. Notochord is solid, again on the dorsal side. These are the gill slits, pharyngeal gill slits, and this is the post-anal tail. So post-anal tail and a closed circulatory system is also a feature here. See the stable comparison. Notochord present, notochord absent. CNS dorsal hollow single, CNS ventral solid double. Pharyngeal gill slits, pharyngeal absent. Heart ventral, heart if present it will be dorsal, and post-anal tail present, post-anal tail absent. Now chordata is further divided into 3 subphyla. See, check it out, subphyla. Urochordata, also known as tunicata, cephalochordata, vertebrata. Now, eudochordata and cephalochordata, collectively known as protochordates. Fourth, exclusively marine. Exclusively marine. Then they will be urochordata and urochordata and cephalochordata. So we have number one, exclusively marine, we have Ctenophora, we have Echinodermata, we have Hemichordata, then Protochordata, and in Protochordata write down Euro as well as Cephalochordata, right? 4. In Euro, only notochord only in the larval tail. Cephalochordata extends from head to tail and persistent throughout the life. Urochordata example, Acidia, Salpa, Doliolum, and even Herdmania. Cephalochordata branchiostoma known as Amphioxus and lancelet, right? Now the members of vertebrata possess notochord during the embryonic stage only. Why do we call them vertebrata? Because later on the notochord will modify and replaced by a cartilaginous or bony vertebral column in the adult. Very important line which you will never forget in your whole life. All vertebrates are chordates, but all chordates are not vertebrates, right? Now see this table. So phylum chordata, subphylum vertebrata, then division. So this is the subphylum, this is the subphylum, yes. This is the division, 2 divisions. Agnatha means without jaws, Gnathostoma means with jaws. Then this has one class, Cyclostomata, right? And it has 2 superclasses based on the locomotory structures. What is the basis of classification? Locomotory structures here, right? If the locomotory structures are fins, we call them pisces and the true fishes. And if they have 2 pairs of limbs or 4 limbs, we call them tetrapoda. Tetra means 4. Now, true fishes, these are the true fishes, pisces. So they bear fins. If their skeleton is made of cartilage, it is chondrichthyes. If it is bony, it is called osteichthyes. So cartilaginous and bony fishes respectively. And then they bear 4 limbs. 2 pairs, amphibian, reptilia, aves, and mammals. Is this point clear? Now, talking about cyclostomata, all living beings or members of the class are ectoparasites on some fishes. So the word is all here. Word is all here. So how many pairs of gill slits? 6 to 15 pairs. Another exercise that we are going to do on your book, maybe on this side or separately on your copy, wherever. Make 3 columns. We are going to write down the name of the number of the gills. One in cyclostoma, then in chondrichthyes, and then in osteichthyes. See here, it has 6 to 15 pairs of gill slits for respiration. Sucking circular suctorial mouth without jaws, devoid of scales and paired fins. So scales as well as paired fins both are absent. So understand the line carefully. So cranium and vertebral column both cartilaginous, circulation closed site, they show migration. Basically they are marine, but they will migrate to the fresh water. Is that clear? Basically all of them are marine, but they will migrate to the fresh water for spawning, right? And after spawning they will die, and only the larva after metamorphosis will come back to its mainland, that is ocean. So example is Petromyzon lemprae, Mexine is hagfish. This is already a PYQ. Is that clear? Next, moving on to chondrichthyes here. These are chondrichthyes, they are marine. Chondrichthyes are marine, right? So write down this also, marine. Cartilaginous endoskeleton, mouth located ventrally downwards, notochord persistent and cartilaginous, without operculum, placoid scales, teeth modified placoid scales, backwardly directed, very powerful, absence of air bladder, so has to constantly swim to avoid swimming. Now examples, electric ray, torpedo, poison stingray, that is trigon. They're all fishes, be it cartilaginous or bony, they are all cold-blooded. Males will have in males, pelvic fins will bear claspers. So these claspers are present near the pelvic fins and they help in holding the female during copulation. They show internal fertilization and many of them are viviparous. Is that clear? Scoliodon, Pristis, Carcodon and the trigon. You should know their common names as well. Then talking about osteichthyes, both marine and freshwater. So again, you've got your answers, right? Exclusively marine. These ones are both marine and freshwater. Mouth, terminal, 4 pairs. See how many pairs of gill slits are present here? Sorry, here 6 to 15. 6 to 15. Is that clear? How many are present in the chondrichthyes? Write down that. And how many are present in the osteichthyes? You should write down that. 4 pairs of gill slits are present in the operculum. Covered by, operculum. Operculum is absent in there. These ones have placoid scales. These one will have cycloid and ctenoid scales. Air bladder absent, air bladder present. Heart is 2-chambered in both. One auricle, one ventral. Both are cold-blooded, told you. Sexes are separate in both. Fertilization internal, fertilization external, usually. Mostly viviparous, mostly oviparous. Development is direct. Now learn the examples: marine, exocetus and hippocampus, and freshwater, labio and catla and clarius, and aquarium, beta and pterophyllum. These names, all these are this aquarium, pterophyllum and beta, they are PYQs. Then talking about amphibian, body divided into head and trunk, skin moist without scales, eyes have eyelids, tympanum represents ear, and alimentary canal, urinary canal and reproductive tracts open into common chamber called cloaca. It was questioned in this year as well. Gills, lungs and skin involved in respiration, cold-blooded, fertilization external, sexual dimorphism will be there, sexes will be separate, and we see that in frog as well. Oviparous, and development is indirect. There is a larval stage involved, right? Bufo is toad, rana is frog, hyla is tree frog, salamandra is salamander, ichthyops, the limbless amphibian, looks more like a very slippery snake, right? That is ichthyops. Now, so coming on to reptilia, so body covered with dry and cornified skin. Is it clear? Epidermal scales and scutes, crawling and creeping type, mostly terrestrial. Again, tympanum represents the ear. Limbs in 2 chambers, 2 pairs, 4-chambered heart only in crocodiles. Otherwise, they will also have usually 3-chambered heart. Usually 3-chambered heart, but 4-chambered heart in crocodile, alligator and gavialis. Not only crocodile, crocodile family. So crocodile, gavialis and alligator all will have 4-chambered heart. Cold-blooded, snakes and lizards lose their skin as skin cast. Fertilization internal, sexes separate, oviparous, development direct, no larval stage involved. Is that clear? Then see the examples here. Yeah, see the examples here. Chelone, turtle, testudo, tortoise, chameleon is the tree lizard, calotis is the garden lizard, crocodilus, alligator, see, and gavialis. All three of them, and one is gavialis, ghadial. All three of them will have 4-chambered heart. Is that clear? Then hemidactylus, this is our friendly wall lizard. Poisonous snake, naja is cobra, bangarus, crate, and viper. Cobra is poisonous. Is that clear? Naja. While boa, ajgar, that is not poisonous. Is that clear? Now talking about avis. So avis is the bird's presence of feathers in all except flightless birds. Which are the flightless? Ostrich, that is truthio, penguin, kiwi, you know. Now they have beak. The mouth is modified into beak. They don't have teeth here. Forelimbs modified into wings. Hind limbs generally have scales for walking, swimming, clasping the tree branches. Skin is dry, without gland, except the oil gland at the base of the tail. Endoskeleton fully ossified. Ossified means bony, but the bones have air cavities. Bones are hollow so that there can be air stored, and air will keep the body light. And pneumatic bones, right? Digestive tract has 2 additional chambers, crop and gizzard. Now crop is always for storing, gizzard is always for churning the food. Warm-blooded, homeotherms, able to keep the body temperature constant. So there is double respiration also, not only double circulation, they should double respiration also because of air sacs connected to lungs to supplement the respiration. Is that clear? Sexes separate, fertilization internal, gives eggs, oviparous, and development direct. See the examples. Corvus, crow, columba, Psittacula, struthio, pavo, Atenoditis, that is penguin, and neophron. Now mammalia. Most unique mammalian characteristic is the presence of mammary glands. Is it clear? To nourish the young ones. They have 2 pairs of limbs but adapted for climbing. Some of them can have variations in that. Then mammals, unique feature is hair, right? Now pinna may not be present in all, like dolphins don't have pinna. You will not see pinna in case of whales also, but pinna will be present only in mammals. Is that clear? Different type of teeth, that is heterodont, what am I saying? So heart 4-chambered, they are homeothermal, that is warm-blooded again. Respiration by lungs, separate sexes, and fertilization is internal. Viviparous, development is direct. So see the examples. Which one are oviparous? We have 2 examples in oviparous mammals, Ornithorhynchus and another is Echidna, right? Viviparous, Macropus, kangaroo, pteropus, flying fox, Camellius is camel, macaca, monkey, rattus, canis, felis, elephas, equus, delphinus, Baliantara, then Panthera, leo, and tigris. So these are the examples of mammals. Please tell me guys that all these points are clear. All the serious guys, all the serious ones. Yes, please. Time for you to respond now. Time for you to respond. Yes, guys. Is that clear? And guys, please don't forget to see, let's take a break. In the chat section, there is a link given. Please click on that link and enroll for the All India mock test right now. Let's take a break. By that time, I'll open the other PDF. Quickly click on the link and enroll for the AIMT. Yes, guys, please. Quickly do that. Quickly do that. We're going to start with next. Unit, that is structural organization in plants and animals. By the way, I'll be reading the introduction and about these scientists separately in a different session. So all things are planned, by the way. So don't worry about anything. Just stay tuned. You have to keep revising things with me. So we are going to start with next chapter, guys. That is morphology in flowering plants. Yes, plus. Have you all uh, enrolled for the AIMT, everyone? Guys, see, I have already completed marathons with all of you, right? And now today I'll be completing class 11 biology. Tomorrow I'll be completing class 12 biology. Done? And after that, I will be discussing some most important topics based on the PYQs, right? And we will be discussing the most expected questions also based on what has already come up in 3rd May exam. So we will be discussing all these things one by one. So there not be a single day when we don't have a session. If by chance not on YT, because other educators have also planned their own sessions, then in that case I'll be seeing you on in special classes on Unacademy Learners app. So make sure that you follow me everywhere on the app as well as on the YT, so that we can stay connected and we are able to revise things every day. Revision is the key. Revision is the key. Practice is the most important. Okay. Shall we move on now? Shall we start? Yes, please. Enrolled, Kirtana? Very good. Who else has enrolled? Mahesh has done that. Who else has enrolled? Quickly give me a thumbs up that all those of you have enrolled for the All India mock test. Yes, guys, once again, give me a thumbs up. Hari Priya, Anjana, Mahesh, Joshila, Sri, Anurag, study, Ashi, very good, very good. Okay, Ashok, cool. All of you have done that? Perfect, perfect, perfect. Let's move on to the next chapter, that is morphology. Chapter full of examples. So we have lots of mnemonics here. I hope. Also, let me know how many of you are taking down these mnemonics. I think you have attended my marathons as well. I've told you then also. I'm repeating them today as well. I'll keep on repeating till your Re-NEET exam. And those who are going to take 2027, if by chance you are sitting here and listening, I'll keep on repeating the same things till you learn, because the whole motive is to learn. You know, sometimes we just learn by listening to things repeatedly. You have to repeat them yourself. And you know what? Always repeat things loudly. What you want to revise and what you want to remember, speak it out loud. Let your ears listen to you, right? It is not always that you have to listen to someone else. You have to listen to yourself also, your heart, your voice. So listen. So speak it out loud. Now, talking about morpho, what is morphology? Morphology is the study of external structures. Talking about root, that it is formed in dicots, guys. The root is formed by direct elongation of radical, and this root from radical forms the primary root, also known as the tap root. Is that clear? Now see here. So primary root system will form the tap root system, as in mustard. So we know that mustard is a dicot. In monocots, the primary root is short-lived and it is replaced by a large number of roots which will originate from the base of the stem, right? And they constitute the fibrous root system, as in case of wheat. In some plants like grass, monstera, banyan, roots arise from parts of the plant other than radicle. If it is arising from parts other than radicle, then it is known as adventitious roots, and they generally have supporting function. Is it clear? What are the general functions? Absorption, providing proper anchorage, storage, and plant growth regulators are also secreted. Is that clear? Now, coming on to count. Regions of the root. So there is a thimble-like structure called root cap, which protects the tender, means soft apex of the root. Now there is region of meristem. So this is the root cap. Above the root cap, we have a region of meristem, then region of elongation, and finally the region of maturation. So region of meristem. Meristem means they will have meristematic cells, actively dividing cells. They are small, thin-walled and dense protoplasm, divide rapidly. Then proximal to this undergo rapid elongation and enlargement and forms the zone of elongation. This region is responsible for actually growth in length. So maximum growth in length of the root is because of this zone of elongation. And then proximal to the region of elongation is the region of maturation, where very fine, delicate thread-like structures called root hairs arise, and they are meant for absorption of minerals. See, rhizoids, like in bryophytes, they are not meant for absorption. Root hairs will absorb water. Is that clear? Now, talking about stem, ascending part of the axis. Ascending means which grows upward, right? Then arises from the plumule, bears nodes and internodes. Also, it bears buds which can be terminal or axillary. Stem is green when young and later becomes woody and dark brown. It spreads out branches, functions, conducts water, minerals, photosynthetates. Photosynthetates means the product of photosynthesis, right? And also storage of food, support, protection, and vegetative propagation. Let's talk about leaf. Leaf is lateral, green, generally flattened structure, develops at the node, bears a bud in the axil, originates from the shoot apical meristem in the acropetal manner. Acropetal means new towards the tip. Is that clear? And organs of photosynthesis, right? Now, talking about the typical leaf. So, a typical leaf consists of three main parts. What is leaf base, petiole, and lamina? Leaf base, petiole is the stalk, and lamina is this broad structure. Now see, the leaf base can have this small leaf-like structure, and two in number. So two lateral small leaf-like structures at the base are called stipules. In case of monocots, guys, leaf base will expand to form the leaf sheath surrounding the stem, right? Partially or wholly. In legumes, the leaf base may be swollen and it is known as pulvinous. And in petiole, it helps hold the blade to light. What is the function of the stalk? The petiole holds the blade to light and it allows the leaf blade to flutter in wind for the cooling and bringing the fresh air, right? Now talking about the lamina or the leaf blade, it is that expanded part which is all full of veins and veinlets. Middle prominent vein, right? The middle prominent vein, this one which is known as the midrib. Veins will provide as a skeleton also, so provide rigidity and also act as a channel for transport. It is a pipeline system, right? For water, minerals and food material. The shape, margin, apex, that will differ. Now talking about venation here. So venation is the arrangement of veins and veinlets. So dicots will have reticulate venation. See, there is main vein, then these veins, then smaller veins, veinlets. Why? So this is a characteristic of dicot leaf. And there is parallel venation in case of mostly monocots. So see the parallel venation here. See the parallel venation. So this is the midrib. Rest all are running parallel to each other and to this midrib. Right, this is parallel and this is a characteristic of monocots. Now types of leaves. So we have two types, simple and compound. 2 points that you have to remember about simple leaf. Lamina is entire. Is it clear? So what you saw here, this is the entire lamina. Is it clear? Around this midrib, this is the entire lamina. Now see. And if it is incised, means if there are cuts, then the cuts do not touch the midrib. The cuts will not touch the midrib. So even if there are cuts, the cuts will not reach the midrib. So midrib is far away from these cuts. Are these points clear? Now, talking about the incisions of the lamina reached, if the incisions reached to the midrib like this, so it was supposed to be one leaf. It was supposed to be one leaf. But what happened? These got cut and the cuts reached the midrib. They reach the midrib, and that is how the leaflets got formed. Is that clear? So incisions of lamina reach to the midrib, breaking it into number of leaflets. This is the compound. And bud is present in the axil of petiole of both simple and compound, but not in the axil of leaflets. So see here. So bud will be present here. So this is the bud. This is the bud. So the whole structure ahead of this bud, this represents a complete leaf. But this one leaf is formed of small leaflet-like structures, small structures. So hence it is a compound leaf. Is this clear? So it is saying bud is present in the axil of petiole. Here, not here, not here. Are you getting these points? Not in the axil of leaflets. Clear to everyone? Now, compound leaves can be of two types, pinnately compound and palmately compound. See here, the leaflets present on a common axis, that is rachis, represents the midrib. In neem, that is pinnately compound, and in palmately, leaflets are attached to the common point at the tip of the petiole. Do you see the structure here? So this is the midrib, which is known as the rachis here, and leaflets are present around the rachis. While in palmately, that is in case of silk cotton, all the leaflets are present around the tip of the petiole. So this is the tip, and this is the this is the petiole, this is the tip. All are surrounded around the tip of the petiole. So it looks like a palm of the hand, hence palmately compound. Are we clear? Please tell me, are we clear? Now, next is phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves. Now see, we have three types of phyllotaxy. In alternate, single leaf at each node. Single leaf at each node. Is that clear? For example, in the alternate manner, China rose, mustard and sunflower. So, what is the mnemonic for alternate? For alternate, it is China, must, sun. China, must, sun or sun. Sun means to hear. So China, must, sun, China flower, mustard and sunflower. For opposite guys, for opposite we have Kala Gora. So Kala Gora is black and white. They are automatically opposite to each other. So Kala for calotropis, Gora for guava. And for world, where there are more than two leaves from each node, this is Elstonia. Is that clear? Is that clear to all? So guys, next is inflorescence. Let's hear more about inflorescence. So flower is a modified shoot. Apical stem changes to floral meristem. Internodes do not elongate. Axis gets condensed. Shoot tip transforms into a flower. And if shoot tip transforms into a flower, it is always a solitary flower. The arrangement of flowers on the floral axis is known as inflorescence, right? There are two types of inflorescence, racemose and cymose. In racemose, the main axis continues to grow and flowers are born in laterally in the acropetal succession. So racemose will have acropetal succession, and in cymose, the axis will terminate into the flower. So the new flowers will arise from the base. So limited growth, and cymose will always have basipetal arrangement of flowers. Means the new at the base. So that is cymose. B and C come together. So remember, basipetal in case of cymose. Yes guys, are these points clear to all? Are these points clear to all? Please tell me. Alright, now talking about the flower, a typical flower will have different kinds of whorls. Swollen end of the stalk. The stalk of the flower is known as pedicel. The tip of the pedicel will be swollen to form the thalamus or the receptacle, where all these whorls are present. Calyx, that is sepal, corolla, petals, right? Calyx and corolla accessory, androecium, gynoecium are reproductive, right? Calyx, corolla, when not distinct, they are known as perianth. Is that clear? Now, if a flower will have both androecium, gynoecium, it will be bisexual. If it has only stamen or only pistil, it will be unisexual. Now see, we have examples here again. So radial symmetry, as in case of animals, the radial symmetry in plants, we call it actinomorphic. So radial symmetry, actinomorphic. Zygomorphic is the bilateral symmetry. Now again, we have to remember the examples. See the examples here. For actinomorphic, I say Dhatura must act chill. Dhatura must act for actinomorphic and chill. Is that true? So that's how we remember. So Dhatura must act chill, and for zygomorphic, that is bilateral, it is Kesia ne Gulmohar P K Bean Bajai. Is that clear? Can you just remember that? Okay. Ziggy Kesia. So zygomorphic Kesia Gulmohar P K Bean Bajai. So this is the mnemonic, right? And for asymmetrical, which cannot be divided into two similar halves, that is Kana. Please tell me these points are clear. Then, if the whorls are in the 3 or in the multiple of 3, we call it trimerous. If it is in the number, if it is 4 in number or the multiples of 4, tetramerous. 5 in number or multiples of 5, it is pentamerous. Is that clear? Then, there are structures called bracts. What are bracts? So, bracts are the reduced leaf-like structures found at the base of pedicel. That is the stalk of the flower. Is that clear? So, if a flower has bract, it is bracteate. If it doesn't have, it is ebracteate. Are these points clear? The diagrams are very, very important, and we are going to come back to this in a different session. Very important. Then, another very important paragraph. So, guys, we have 3 types of flowers based on the position of the ovary. See, based on the position of calyx, corolla, and androecium with respect to the ovary on the thalamus. So this is the basis. We have 3 types of flowers. One is hypogynous, second is perigynous, and third is epigynous, right? Hypogynous, hypo means below, gynous means ovary. So hypogynous means gynoecium will be the highest in position and everything else will be present below it. And in such case, if ovary is at the highest position, that ovary is called superior. Is that clear? So we have mustard, china rose, and brinjal. Then in gynoecium we have in perigynous, gynoecium is in the center and all other parts are on the periphery. Perigynous, peri means around the gyne on the rim of the thalamus. So everything is at the same level actually. So honestly, it depends upon the structure of the thalamus. Is that clear? So gynoecium in the center and everything on the same level, on the rim of thalamus on the same level, written. So this is perigynous. So such ovary is also not superior, but we call it not even inferior, half inferior. So example is plum, peach, rose. Epigynous above ovary. So ovary will be at the lowest position. So lowest ovary is called inferior. Other parts arise above ovary. Is that clear? So basically thalamus grows upward, enclosing the ovary completely. And example guava, cucumber, and ray florets of sunflower. Are we clear till here? Yes, guys, are we clear? Now let's talk about flower. Calyx is a collective term for all sepals. So if sepals are fused or united, the term is gemosepalous. Sepals are free, we call it polysepalous, right? Then similarly gemopetalous and polypetalous. Is that clear? Then estivation. Now see the definition here. Arrangement of sepals or petals in the floral bud with respect to each other, like actually to the members of the same whorl. So petal to petal has to be compared. Sepal to sepal has to be compared. So it is an arrangement with respect to other members of the same whorl. So please, while you are reading the statement, by chance, if they ask you this question, statement based question, so while reading the definition, focus on this word. It is the arrangement of sepal petal in the floral bud with respect to other members of the same whorl. It doesn't have to be sepal or petal being compared. Is that clear? Now the main types of estivation, guys: valvate, twisted, imbricate, right, and vexillary. Now when sepals and petals in the whorl just touch one another at margin, no overlapping at all, this is valvate. Margin to margin, no overlapping, valvate. Margin overlaps the other one. One margin overlaps the next one. One is inside, other is outside. that way, if the margin is present, that is called twisted. So see the example, valvate in calotropis, twisted in china, rose, ladyfinger and cotton. Then in imbricate, there is no fixed pattern. So either both the margins can be enclosed by the other petal, or both the margins of a petal or the sepal, they are on the outer side. So there is no fixed pattern, not in a particular direction. That is imbricate, GIC. Gulmohar, I for imbricate, and cassia. Is that clear? Then we have fourth type, that is vexillary, also known as papilionaceous. In this, there is a fixed pattern. Fixed pattern is that there are always 5 petals. Out of these 5 petals, one is the largest one, which is known as standard, and it is on the posterior side. Is that clear? The largest one is on the posterior side. Then this standard overlaps the 2 lateral petals. They are smaller as compared to the standard. They are called wings, which in turn, now the petals, wings, they will in turn overlap the 2 smallest ones, and the smallest ones are known as keel, and these keels are fused, and the keel one is on the anterior side. So you have to take notice of many words here. The largest one, standard, overlap the 2 lateral ones known as wings, which are smaller, and then the wings will further overlap the keel. Now the keels are also fused. Lateral wings are not fused, keels are fused. Is that clear? Standard is on the posterior side and keel is on the anterior side. So if you ever go through the questions on this topic, you will always find them writing that keel is on the posterior side and standard is on the anterior side. So the statement goes wrong. Is that clear? So watch these words. And why do we call it papilionaceous? Because it is found in one of the subfamilies called Papilionaceae. So hence, because it is a characteristic feature of subfamily Papilionaceae, which includes pea and beans. Hence the name papilionaceous. Is that clear to all? Is that clear to all? Right? Then talking about androecium here. So see, androecium represents the stamens. So this is a male reproductive organ, has stalk known as filament and anther. Anther is usually bilobed, which has 2 chambers known as pollen sacs. Means each lobe will have. So overall there will be 4 pollen sacs. So tetrasporangiate, right? Pollen grains will be produced in the pollen sacs. Sterile stamen, which will not produce pollens, that will be known as staminode, right? Now, if stamens are fused with the petal, that condition is called epipetalous, like in brinjal. If there is no petal sepal distinction, and instead of petal and sepal, the flower has tepals, and in case stamen is fused with the tepal, then we call it epiphyllous. Collection of tepal is known as perianth, right? So epiphyllous when attached to the perianth, like in case of lily. These examples are very important. Now sometimes the stamens are separate, they are free. Then if they are free, we call it polyandrous. If they are fused, we call it synandrous. Is that clear? So this is free, polyandrous. If they are fused, then we use the term synandrous. Syn means fusion. Now synandrous can be monoadelphous if fused to form 1 bundle, diadelphous fused to form 2 bundles, and polyadelphous fused to form more than 2 bundles. So you must know monoadelphous, mono, MM should be common. Monoadelphous is seen in case of Malvaceae. China rose is a member of Malvaceae. Is that clear? Diadelphous in Fabaceae, pea, beans, right? And polyadelphous in case of citrus. Is that clear? Then all the stamens of within a flower may not be of equal length. If there is variation in the lengths of anthers, then that can be seen in salvia and mustard. That can be seen in case of salvia and mustard. Are we clear? Are we clear? Now talking about gynoecium. See here. Okay. So we know gynoecium is a female reproductive part, it consists of many carpels. Each carpel has 3 parts, stigma, style and ovary. right? Now, ovary is a large basal part, style connects ovary to stigma, stigma at the tip, and then ovary bears one or more ovules attached to a flattened cushion-like structure, called placenta, and when more than one carpel is present, so when carpels are free, okay, so that is called apocarpus. So free are apocarpus seen in lotus and rose. If there are more than one and they are all fused, this is called syncarpus. This is seen in case of mustard and tomato. Is that clear? Now talking about this very important topic called placentation. So this is the arrangement of ovules. So now watch these words carefully. In marginal, guys, placenta forms a ridge on the ventral suture. The ovules are born on this ridge in two rows, like in P. So marginal and P. So marginal placentation is a characteristic of P. So when placenta is axial, ovules in multilocular ovary, placentation is axial. China rose, tomato and lemon. In parietal, guys, ovules develop on inner wall of ovary. Ovary one chambered becomes two chambered due to false septum. So false septum is a character of parietal. Multilocular ovary in case of axial, and true septum is present here. Is that clear? So underline these terms. False septum is a character of parietal, and then there is one chamber. In axial there is a multilocular ovary, and there is true septum, which will divide it into chambers. Is that clear? Example is mustard and argemone. Then septum absent in case of free central. Example is dianthus and primrose. In basil, there is only one ovule at the base of the ovary. Example sunflower and marigold. Are these points clear? So these are the structures you should be able to identify. Now guys, talking about fruit. So fruit, characteristic feature of flowering plants, yes. Now true fruit is formed by ripened ovary, while a false fruit will be developed from any part other than ovary, usually thalamus. Then we have a fruit which develops without fertilization, which is known as parthenocarpic. So if there is no fertilization, it will be seedless as well, right? So generally the fruit consists of a wall. What is a fruit wall known as? Pericarp. How does it develop? From the ovary wall, right? So pericarp and the seeds. Now this pericarp can be dry or it can be fleshy. Now see, this pericarp can be thick and fleshy and differentiated into three layers: epicarp, mesocarp and the endocarp. Now talking about the fruit type in mango and coconut. Mango is a dicot, coconut is a monocot, but still the fruit type is same, known as drupe. So we call drupe to such fruits. Drupe is a name or the term given to such fruits which have these characteristics. See, they develop from a monocarpellary condition. Monocarpellary, superior ovary and one seeded. That is a rule for drupe. Monocarpellary, superior and one seeded. Is that clear? So such fruit is known as drupe. So see here, in drupe, let's see the arrangement and the nature of three layers. So see, epicarp is outer and thin. Here also and here also, but here since they are using a mature coconut here, so we are not able to see epicarp here. Otherwise, the coconut we drink coconut milk from, that green cover, the green outer layer, that represents the epicarp. Okay, then in case of mango, the edible part, the fleshy, juicy, fibrous part that we eat, that is mesocarp. And in case of coconut, these brown, fibers, sclerenchymatous, thick structures, these are mesocarp. But again, in both the cases, they are fibrous. And then the outer layer, the outer stony, hard endocarp. And the same is in the case of coconut as well. Is it clear? So outer thin epicarp, middle fleshy edible mesocarp and inner stony endocarp. In coconut, the mesocarp is fibrous. Is it clear? Not fleshy but fibrous. Is that clear? All right. Let's talk more about seed. Now seed consists of seed coat, testa, tegmen and embryo. And embryo is further made up of radical, plumule and radical, embryonal axis and one or two cotyledons. Is that clear? So 1 cotyledon in monocots and 2 in case of dicots. Let's see the structure of dicot seed here. Outer testa, inner tegmen, right? So how do these seed coats develop? From the integuments of ovule, right? Testa, tegmen. Hilum is a scar on the seed coat through which the developing seeds were attached to the fruit. Clear? Now, small pore known as micropyle that remains. What is the function of micropyle? That is for absorption of water and gaseous exchange. Cotyledons are seed leaves. They're full of food reserve, right? Then, in some seeds like castor, the endosperm formed as a result of double fertilization in food storing called endospermic seed. Now, what is it trying to tell you? See, basically we know that dicot seeds are usually non-endospermic. Why? Not because endosperm was not formed, but endosperm formed was completely consumed during the development, right? So normally and usually dicot seeds will use up, they will consume their whole endosperm. But there is this example castor. It is a dicot in which endosperm is not completely consumed. So it's an exception amongst dicots which has endospermic seed. Please learn it well. Okay. Otherwise, mostly like pea, bean, gram, these are dicots, all dicots. Castor is also dicot, but these will not have endosperm remaining behind, but castor will contain the remnant of endosperm, hence endospermic, and while these are non-endospermic, and typically dicot is non-endospermic. Is that clear? While monocot seeds, they are endospermic because the endosperm is not completely consumed, while orchid is also a monocot, but in which endosperm is completely consumed, hence it becomes non-endospermic. Can you remember these things? Yes, guys? All right, now just see the structure and the labeling of this structure. So this is a monocot seed, basically a maize. So the outer layer is a seed coat and fruit wall consumed, major part made up of yellowish endosperm, and it has this scutellum, the monocot single cotyledon. This is the embryonal axis, plumule covered by the sheath coleoptile, radicle covered by the sheath coleorhiza. Coleorhiza and coleoptile are absent in case of dicots, right? So see, what is endosperm here? Endosperm is bulky and stores food. Proteinaceous layer made up of aileron layer. Embryo small and situated towards a groove, and a shield-shaped cotyledon known as scutellum. Short axis with plumule and radicle. Plumule and radicle enclosed within the sheaths, which are absent in dicot. And then, guys, we have this typical flowering plant. Some technical description known as floral formula, and then we have a typical diagram, floral diagram. See here, what are the abbreviations we use for floral formula? BR for bracteate condition, K for calyx, C for corolla, P for perianth in case tepals are present and not calyx and corolla, A for intrusium, G for gynoecium. Now, if G is above the bar, if there is a dash, if there is a bar below, so G is above the bar, it is superior. If G is below the bar, it is inferior. Okay, now this is the sign for male, this is the sign for female. If we combine two, it becomes a bisexual flower. A plus sign indicates that a flower can be divided in any plane to get two similar halves. This is actinomorphic. Then we have zygomorphic. The sign is that of percentage. So there is just one plane in which you can get two similar halves. Now fusion. Fusion means when the structures within the same whorl are bound to each other. So this is indicated by a bracket, while adhesion means when two different whorls are connected to each other. For example, epiphyllus, epipetalus. So stamen attached to perianth or stamen attached to petal, that is called adhesion. While fusion is basically cohesion, when the elements of the same whorl are bound to each other, are fused. That is cohesion. Co means similar. Is that clear? Now, cohesion and adhesion within the parts can also be shown through floral formula, right? Floral diagram represents, this is the floral diagram of the mustard or Brassicaceae family, which is in your syllabus though. Is that clear? Now, So this is the potato family, guys. We'll be talking about the families in a separate session. So with this, we complete the chapter of morphology. Now coming on to anatomy. Yes, guys, quickly give me a thumbs up that you are thorough with what we have discussed so far. Quickly let me know. My tab has gone off. Let me, oh, wait. Just a second, guys. Let's take a quick break, just 2 minutes, and come back. Just 2 minutes. Yes, guys. So break is over. Break is over. Let's start with the next chapter, guys. Let's start with the next chapter, which is anatomy. Are we all here? Give me a power thumbs up that we are all here, not tired. Yes, guys. All here with me? Shall we start with the next chapter? Yes? All right. Ooh. All ready? Syngenesis and synandrous. Anjana, syngenesis and synandrous condition. Okay. See, synandrous we know. Yes, please. Anything else? I'll explain. See, there's a difference. In synandrous, what happens? In synandrous, all the stamens are fused together. Okay? They are not joined with any other part. It is only the stamen all bundled up together to form one bundle, one cylinder, or maybe two groups. That is synandrous. Okay? And then you're asking syngenesis. Yes, in syngenesis, what happens? Anthers are fused, but around the like in style around the style. In case of Malvaceae, China rose, do you see it is a hollow style, and around that hollow style all the stamens are fused together in one bundle. That is syngenesis. Do you see the difference? So stamens are around the hollow style. Any other question here? While the filaments, they are free from each other, but they are around the same style. That is syngenesis. Clear? What else? All right. All clear? Please let me know that we are all set and we can start with the next chapter, guys. Quick, quick, quick. All the serious students? Yes, please. All the serious students here. All right. Let's move on to the next chapter, that is anatomy in flowering plants. So study of internal structure of plants is known as anatomy. Internal structures will show adaptation to the diverse environment. Talking about the tissue system, there are three tissue systems. One is the epidermal tissue system, then the ground tissue system, and the vascular or the conducting. Talking about the first epidermal tissue system, outermost it will form the covering and consist of epidermal cells, stomata, epidermal appendages, which are either root hairs or stem hairs called trichomes. Epidermis, outermost layer of the any primary body part, be it stem, root or leaf, the outer layer will be known as epidermis, right? Made up of elongated, compactly arranged, forms a continuous layer, usually single layered except in case of xerophytes, where it is a xerophytic adaptation. So multi-layered epidermis can be seen in that, but usually it is saying single layered. What kind of cells are present? Parenchymatous. They are continuously present there where without any intercellular spaces. It has a large vacuole and waxy thick layer called cuticle, which prevents the loss of water. But remember, cuticle is absent in root. If a waxy layer is present around the root, how will water be absorbed, right? So roots do not have cuticle. It is absent in roots. Remember that, right? Absent in roots. Stomata present in the epidermis of leaves as well as on young stem, right? Then each stoma has two bean-shaped cells known as guard cells, and the opening between them, pore between the guard cells, is known as stoma or stomata. In monocots, the shape of the guard cells will be dumbbell shaped. See the diagram. See this diagram. In monocots, the shape of the shape of the guard cells is dumbbell shape, while here it is bean shaped, right? Now, very important, and listen carefully, mark these words carefully, read them carefully when you get your paper. So outer wall of the guard cell is thin, right? And the inner wall towards the stomatal pore, do you see a bold line here? So the inner walls are highly thickened while the outer ones are thin. Is that clear? Now, few epidermal cells in the vicinity of guard cells. So do you see these guard cells and these dark green colors? These are called subsidiary cells. These are also modification of the epidermal cells. Now, three things: subsidiary cell, guard cell, and the stomatal pore together forms the stomatal apparatus. Are these clear? You should be able to label the diagrams properly, and also we have got this diagram in our PYQ. Now see, the cells of epidermis bear number of hairs. The root hairs are unicellular. Root hairs are always unicellular, unbranched, while the trichomes, that is the stem hair in the shoot system, usually multicellular, but not always, right? These are always unicellular, these are usually multicellular. Always unbranched, these can be branched or unbranched. These are meant for increasing the surface area to absorb water, while these are secretory and prevent the water loss due to transpiration. Is that clear? Then coming on to the ground tissue system. It will consist of parenchyma, collenchyma, sclerenchyma, all of it. Now, parenchymatous cells are usually seen. It is the word usually. See, the word usually makes the statements generally right. If you get a statement with words like only, always, you should be little careful while reading such statements, right? So parenchymatous usually present in cortex, pericycle, pith and medullary rays, in the primary stems and roots. Leaves, the ground tissue, consist of thin-walled chloroplast containing cells and is called mesophyll. Is that clear? All right. Now, coming on to the third type of tissue system, this is a vascular tissue system. Dicots have cambium. Be it dicot stem, be it dicot root, it will have cambium. Cambium is meristematic in nature. So cambium will help in secondary growth. So both dicot root and dicot stem is going to show us secondary growth. And the structure which is open to secondary growth has open vascular bundle. Is that clear? Now you will see the vascular bundle which will have cambium is known as open vascular bundle. See, cambium possesses the ability to form secondary xylem and phloem, hence open vascular bundle. That is in case of dicot. So dicot have open. So how do we remember? Doc we should remember, right? It is a dream. So doc also stands for dicot, open vascular bundle, because of the presence of cambium. Is that clear? Now, in the monocots, the vascular bundles have no cambium, do not form secondary tissue, hence closed vascular bundle. Xylem, phloem arranged in alternate manner. See, xylem, then phloem, xylem, then phloem, xylem, then phloem, alternately on different radius. Alternate manner, different radius. This is known as radial, RR. So there are many RRs in our botany, right? So you have to remember radial arrangement will be seen in root. Is it clear? Always radial. So such arrangement is seen in, be it dicot or monocot root, it will have radial. While in stem we have conjoint. See, in conjoint, xylem and phloem jointly situated along the same radius. So on the outer side we have phloem, on the inner side we have xylem. Is that clear? Now both are conjoined, but here in between xylem phloem we have a layer of cambial tissue, right? So cambium will make it open conjoint, while absence of cambium will make it closed conjoint vascular bundle. Are we clear? So this is conjoint, open, closed, this is radial. Are we clear are we clear? Please tell me. Are we clear, guys? So such vascular bundles common in stem and leaves. Conjoint usually have phloem located on the outer side of xylem. Now let's talk about the anatomy of dicot and monocot plants, starting with dicot root, and they have shown the structure of sunflower root. What does it indicate? That sunflower is a dicot, and we have to talk about sunflower in the Compositae family, right? Compositae family belongs to the dicots. Now, outermost layer is epiblemma. See here, the outermost layer is epiblemma. So epiblemma is the term given to epidermis of root. Then The cells of epiblema are unicellular root hairs. They'll protrude out as unicellular root hairs. Cortex consists of, this is whole cortex, multi-layered, several layers, written thin-walled parenchyma with intercellular spaces. You do see some spaces here, intercellular spaces present. Innermost layer of cortex is always known as endodermis, and it is single layer made up of barrel-shaped cells, Is that clear? Made up of barrel-shaped cells like this. So like this. So these are barrel-shaped cells. This is how the endodermis is present. And then very important thing mentioned. So single layer without any intercellular spaces. While in cortex we do see intercellular spaces, there are no intercellular spaces in endodermis, right? Now tangential and radial walls have water impermeable waxy material suberin in the form of Casparian strips. So there is Casparian strip all around. Now Casparian strip will make it impossible for water to get transported towards the inner layers. So water is getting water is getting absorbed through the root hairs, goes into the epidermis and then cortex. But here, once it comes across the endodermis, the pathway is blocked because of this suberin deposition on tangential as well as on radial walls. These suberin deposition, these layers are known as Casparian strips. Then how does water move into the deeper layers? Then in order to make sure that water transport is not inhibited, that is why the endodermal cells are inhibited. They are interfered by some special cells known as passage cells. So in endodermis, we have some cells which interrupt the endodermal cells known as passage cells to maintain the continuity of water column. Is it clear? Now, when next to endodermis lies a few layers of thick walled. So pericycle is made up of thick walled parenchymatous cells. Is that clear? And it is in few layers here. Now what is the function of pericycle? Initiation of lateral roots, that is the secondary, tertiary roots, they come out through pericycle, and the vascular cambium. Is that clear? Now during the secondary growth, this vascular cambium arises from pericycle, and this cambium will give rise to secondary growth. Now pith is small and inconspicuous. Now look at the diagram. Theory says pith is small and inconspicuous, but if you compare the two diagrams, this appears larger. So please take care of the diagrams. The diagram is fine, but you have to go with the theory because these are NCERT diagrams. Okay, so you might feel that this is larger and you may have that memory of the diagram only, but please focus here. Pith is small and inconspicuous here, while here it appears smaller, but actually in monocot the pith is larger. See here, the pith is larger here. Is that clear? Yes, see here. The pith is small and inconspicuous in dicot. Pith is large and well developed in case of monocot, you have to remember. Now the parenchymatous cells lie between the xylem and phloem called conjunctive tissue. You got this question in 3rd May 2026 paper, right? So what match the following? Do you remember? Match the following we got. So the parenchymatous cells which separate xylem and phloem is called the conjunctive tissue. And now remember, pericycle, vascular bundles and pith, everything inner to endodermis is collectively known as stele. Now the difference between monocot and dicot root is only with respect to two things as far your syllabus is concerned. One is pith. In dicot we have a small and inconspicuous pith. Here we have large and well developed. See here. Here we notice there are only 4 vascular bundles. So at max there can be 4. 2 to 4 vascular bundles are present here. See, 2 to 4 xylem phloem patches are present here. But in case of monocot, guys, there can be more than 6 xylem bundles. So if you count it here, 1, 2, 3, 4, 5, 6, 7, 8. So more than 6 vascular bundles is a character of monocot. So more than 6 is known as polyarc condition. Is that clear? And also cambium is present here. Cambium will develop from pericycle, but here cambium will not develop. So open growth and closed growth. No secondary growth because of no secondary growth because of no cambium. Secondary growth is seen in dicot root because of cambium. Please tell me if the topic of root is clear to everyone. Yes, guys, the topic of root is clear to everyone. Is that clear? All right. Now, moving on to dicot stem. Let's see. So, covered with thin layer of cuticle, it may bear trichomes and fused stomata. Then in dicot stem, the cortex is well developed into 3 subzones: hypodermis of collenchyma, providing mechanical support; cortical layers made up of a thin-walled parenchyma made up of conspicuous intercellular spaces; and endodermis rich in starch are known as starch sheath, right? Pericycle, if we discuss here, pericycle on the inner side of endodermis is made up of semi-lunar patches of sclerenchyma. Radially placed parenchyma in between the vascular bundles. This was also there in 2026 paper. This forms the medullary rays. Now the vascular bundles are arranged in the ring arrangement, and it is a characteristic of dicot stem. What is a vascular bundle like? Conjoined, open, end arc protoxylem. What is end arc protoxylem? Means protoxylem on the inner side towards the pith area, and large intercellular spaces which occupy the central position, they constitute the pith. So pith is very large in dicot stem. Talking about monocot stem, guys. So in monocot, the cortex is not very well developed. Rather, there is a very thin layer known as hypodermis, and that too made up of sclerenchyma. There are scattered vascular bundles surrounded by sclerenchymatous bundle sheath cells. Remember, bundle sheath cells are sclerenchymatous always, right? Large conspicuous parenchymatous ground tissue. So the ground tissue is not differentiated into proper cortex and endodermis and pericycle or pith. They are not present. So it is just undifferentiated ground tissue present, and vascular bundles are conjoined and closed, it being a monocot, right? Peripheral, the outer vascular bundles are smaller and the central ones are larger, right? Phloem parenchyma absent, very important lines, and water containing cavities are present within the vascular bundles. Now talking about the dorsiventral or the dicot leaf. So epidermis on 2 sides, upper one known as addaxial, lower known as abaxial. Abaxial, on the lower side we have more stomata. On addaxial side, upper side, we have cuticle, right? Stomata may be absent on the addaxial side. It is all written. Then mesophyll possesses chloroplast and carries out photosynthesis. The mesophyll is differentiated. I am talking about dicot, dorsiventral. So the dorsal and ventral are differentiated. They can be well distinguished. Now palisade and spongy. Now just below the addaxial, we have palisade, and below the palisade we have spongy. So spongy is towards the abaxial, palisade is towards the addaxial. Are these points clear? Now see, since we know, we have read about venation, in dicots we have reticulate venation. So we have veins and then we have smaller veinlets, right? So see, the size of vascular bundle will depend upon the size of the vein. Larger vein, larger size of the vascular bundle, smaller, smaller. Is that clear? So the layer of thick walled bundle sheath cells are surrounding the vascular bundles. Is that clear? Then talking about monocot leaf. Here isobilateral, both the sides are similar, no distinction. Cuticle on the upper side, cuticle on the lower side. The number of stomata is also similar on the upper and the lower side. So stomata present on both. Mesophyll not differentiated. In grasses, the upper addaxial side has very important cells. See, epidermal cells along the veins modify to form large empty colorless cells known as bulliform cells, also known as motor cells. Why? Because they show movement basically, or help in the movement. Leaves, when they will absorb water, see, bulliform cells, when they absorb water, the leaf surface is exposed, the leaf is open. But when there is a water stress, the leaves will, bulliform cells will lose water. Leaves are going to curl inward to minimize the surface area and to reduce transpiration. Makes sense, right? The parallel venation is reflected in the near similar size of vascular bundles as seen in the vertical section. Are we clear here? Please tell me. So guys, with this, we complete the chapter of anatomy, right? Now moving on to the chapter of structural organization in animals. All right. So guys, I am going to skip this frog because for frog, I've already made a separate video and that will be uploaded very soon. So we are going to discuss frog and cockroach separately in the in a different session. Let's move on to the chapter number 8. All right. Starting with cell structure. Yes, please. Let's talk about cell. Everybody here with me? Anyone who has any doubt? Everybody is here with me. Please tell me. We will take class at least till 4 or 5. Never mind if the session will start. We'll chemistry class. At least till 4. 4 or 4:30 we'll take the class. Okay? All right. I know you have your chemistry class. Don't worry. I'll try not to overlap the classes. In case we are not able to complete, we'll take it in part 2. But we have to do it in a proper way. Yes or no? Let's continue. So unicellular. So otherwise, in case needed, I'll take part 2 of this session. Do not worry about it. Already we have discussed that if light would not have, that this problem of short circuit would not have been sorted, we would have done it tomorrow. We'll do it. Don't worry. Haste, we have to hurry, but we don't have to make so much of haste that it makes waste. Right? So do it properly. Take another hour. We'll take another 2 hours. We still have days. Don't worry. Now, what is cell? Okay. So anything less than a complete structure of cell does not ensure independent living. These lines are clear. Anton von Leeuwenhoek, the first saw and described a live cell. Robert Brown discovered the nucleus. Is it clear? And you know who discovered the cell? It was Robert Hooke. Robert Brown, nucleus. Robert Hooke, cell. And the dead cell. Then Matthias Schleiden, a German botanist, right? And Theodor Schwann, again a German zoologist, right? Both are German. They studied animal and plant cell. Now note it here. So this Schleiden, who was a botanist, he said that all plants are composed of different kinds of cells, right? But just read this carefully. What did Schwann report? Schwann reported that cells have a thin outer layer known as plasma membrane. That is one. Second, he also noticed, because he gave this reading in 1839 and Schleiden gave it in 1838. So he observed it later. So he said that animal cells, see, the presence of cell wall is a unique character of plant cells. Why? He did not observe cell wall in any animal cell. Is it clear? First, he said cells have thin outer layer known as plasma membrane. Second, presence of cell wall is unique only to plant cell. And third, then, see, on this basis, Schwann proposed the hypothesis that bodies of plants and animals are composed of cells and products of cells, right? Schleiden and Schwann together formulated the cell theory, but did not explain how new cells are formed. So that is their disadvantage. And Rudolf Virchow 1855 explained the cells come from the preformed cells, omnis cellula a cellulae. So what does cell theory constitute of? All living beings are composed of cells and the products, and cells arise from pre-existing cells. Are we clear? Now, talking about the overview of the cell here. See, in both prokaryotes and eukaryotes, a semi-fluid matrix called cytoplasm occupies the volume of the cell. Cytoplasm is the main arena of cell activities in both plants and animal cells, even in the prokaryotes, right? And this various chemical reactions occur in it to keep the cell in the living state. And what do we call these chemical reactions as? Metabolic reactions. Metabolism is important for life, right? Now, ribosomes, see, Besides the nucleus, eukaryotic cells have other membrane-bound organelles like ER, GB, then lysosomes, mitochondria, microproteins, and vacuoles. Prokaryotes lack all these membrane-bound organelles. Ribosomes, non-membranous, or you can say membrane-less. They are found in all cells, eukaryotes as well as prokaryotes. In eukaryotes, guys, they will be of 70S type. In prokaryotes, in eukaryotes, they are of 80S and 70S, and in prokaryotes, only of 70S type. Now, see, in eukaryotes, within the cell, ribosomes are found not only in cytoplasm, but also within two organelles, chloroplast and mitochondria, and on rough ER. We are talking about eukaryotes here, right? Now, the ribosomes which are found in cytoplasm, 80S. Ribosomes which are found in chloroplast and mitochondria, 70S. And on ER, again 80S. Are we clear? Right. Animal cells contain another non-membranous or bound organelle, non-membrane bound, yani membrane-less organelle, that is centrosome, which contain two centrioles. So the two non-membrane bound organelles that we know about, one is ribosome, second is centrioles or centrosome. Is that clear? Ribosomes found in both, in fact, all types of cells, but centrosome is found only in animals. Is that clear? Now, cells differ greatly in size, shape, activities. Mycoplasma, we have read, the smallest cell, only 0.3 micron meter, while bacteria could be 3 to 5 micron meter. Largest is the egg of the ostrich, and RBCs 7 micron meter. Longest nerve cell, and then their shapes differ with their function. Now, prokaryotic cell, we have already read the kingdom of Monera, bacteria, blue-green algae, mycoplasma, PPLO, pleuropneumonia-like organisms, right? We have seen the shapes, we have seen overall basic organization of prokaryote and eukaryote is fundamentally similar, right? There is an outer boundary, plasma membrane present in all, right? The basic cytoplasm, which is the main arena for all the cellular activities for both prokaryotes and eukaryotes, the genetic material, but we will see the differences as well. Now see, the semi-fluid matrix fills a cell in is cytoplasm, no well-defined nucleus. We are talking about prokaryotes. Genetic material basically naked. Why? Because of no nuclear membrane. In addition, the genomic DNA, see it, the genomic DNA, many bacteria have small circular DNA outside the genomic DNA, and this extra genomic material is known as plasmid. This plasmid has very important genes which provide them unique phenotypic characters like resistant to antibiotics, right, fertility factor, and so on. Then prokaryotes have something unique in the form of inclusions. Inclusions are not present in eukaryotes. Then there is a specialized structure called a mesosome. Basically, plasma membrane invaginates to form mesosome, essentially the infoldings of cell membrane, only found in prokaryotes. Is that clear? Then talking about the cell envelope and the modifications here. See, prokaryotes have cell envelope, which cell envelope is made up of 3 layers. 3. Outermost glycocalyx, then in between cell wall, and then the plasma membrane. Let me mention here, glycocalyx may or may not be present. Do you remember Griffith's experiment, R strain, S strain? So those which will have glycocalyx will be smooth. And what is the function of glycocalyx? It helps the bacteria to hide from our immune system, right? Cell wall will be present except in case of mycoplasma, and plasma membrane is always present, right? But together they will act as one single unit and give protection. So single protective unit. There are differences in the cell envelope, and on that basis, guys, the bacteria can be differentiated as either gram positive or gram negative, because on the basis of the differences, they take different type of stains. So gram stain, crystal violet. So Christian Gram was a scientist who developed this staining technique and differentiated the bacteria. He realized that this differential staining is because of difference in their structure, right? So those which take up the positive, which take up the gram stain are called gram positive. they appear purplish, right? While those which do not take up the gram stain or actually lose it after washing them in alcohol, they have to be counter stained with red stain and they appear red. So gram negative bacteria, right? So glycocalyx differ in composition and thickness, could be loose sheath. If glycocalyx is loose, slimy, we call it slimer. If it is thick and tough, it forms the capsule. Cell wall determines the shape and provides the structure, and very important, prevents the bacteria from bursting or collapsing depending upon the medium. Plasma membrane always selectively permeable, be it bacteria, be it us, right? Now plasma membrane forms some structures, membranous structures. First of them is mesosome. Due to inward growth or invagination of the plasma membrane is mesosome. This mesosome can be in the form of vesicle or tubule, or it can be lamellar like this. Is that clear? And what are the functions of mesosome? Help in cell wall formation, DNA replication, distribution to the daughter cells, respiration, secretion, increase the surface area, enzymatic content. And then apart from mesosome, there is another inward growth that is called chromatophore. Now chromatophore is only present in the photosynthetic bacteria because chromatophore, chroma means color, so chromatophore contains pigments. Pigments are light absorbing things, right? So it will be only present in the autotrophic ones. Is that clear? Or the photosynthetic ones. Now bacterial flagella consists of three parts: filament, hook, and the basal body. Filament has thread, longest portion. Hook is embedded in the membrane, and is that clear? And it extends from the cell surface to the outside. We are talking about the filament, the thread-like structure. Pili and fimbriae. There are other structures other than flagella. It is pili and fimbriae. See, flagella will have role in movement. Okay, flagella will help the bacteria to move, and there can be wide variety of bacteria depending upon the number of flagella and its distribution on the surface, but it may not always be present. Is that clear? Second, other than flagella, there are structures like pili and fimbriae. Pili and fimbriae don't play any role in movement. Motility, flagella. Pili and fimbriae are for attachment. So the smaller structure is fimbriae, pili is a longer structure. So see, do not play role in motility. Pili, elongated tubular, fimbriae, small bristle-like. Function to attach bacteria to the rocks and also to the host tissue. Is that clear? Also, even in conjugation, they will help. Then ribosomes and inclusion body. See, ribosomes, I told you, membrane-less. Eukaryotes have both 70 and 80S, and prokaryotes will only have 70S. What is the size? 15 by 20 nanometer. So two subunits, 30 and 50S, together form 70S. Role in protein synthesis. So one mRNA thread is covered with multiple ribosomes in case of prokaryotes. So we see a chain of ribosomes on a single mRNA chain. So this aggregation is known as polysome or polyribosome. Now we have read it in genetics. Why does this happen? This increases the efficiency of protein synthesis. So multiple copies of proteins can be formed by this polysome. Is that clear? Then inclusion bodies. Inclusion bodies are only present in prokaryotes. We've read that. So they contain reserve material. Again, membrane less. They lie free in the cytoplasm. So depending upon what do they store, phosphate granules, we'll call them, cyanophycean granules or glycogen granules, and also gas vacuoles. These are four different examples of inclusion bodies. Is that clear? Now, where are these gas vacuoles present? Gas vacuoles will help them to float. They are found in blue-green algae, purple bacteria, green photosynthetic bacteria. Is that clear? Now, talking about the eukaryotes here. See, eukaryotes will have membrane-bound organelles, be it membrane-bound genetic material. Nuclear membrane is present, right? Membrane-bound organelles like mitochondria, ER, GB, lysosomes, we have discussed them. Also, we see this membrane bound thing, basically what are we doing here? We are compartmentalizing the structures. We are developing walls around them. We are developing this extensive compartmentalization because this will help in division of labor. Is that clear? We have different rooms for different things. So organized nucleus with nuclear membrane, genetic material organized into chromosomes. Now for chromosome formation, we want two things. We want that genetic material should be bound with histone proteins. Is that clear? Without histones, DNA will not form nucleosome, it will not form chromosome. So the concept of chromosome is only for eukaryotes. For prokaryotes, we have prochromosome. In prokaryotes, we don't have histones, so no chromosomes. Is that clear to all? Please tell me, right? All eukaryotic cells are not identical, and of course they will differ. In plants, guys, we have cell wall and plastids and large central vacuole, which are absent in animals, while animal cells will have centrioles, which are almost absent in all the plant cells. Clear? Now, we'll come to these structures later. And now, moving on to cell membrane. See, structure of plasma membrane or cell membrane was clear when we had electron microscope. It was only in 1950s that it was discovered, and we could discover the structure of plasma membrane. Now, mainly composed of lipids and proteins, but we also have carbohydrates, but main components are lipids and proteins, fats and proteins. And out of proteins, the major lipids are phospholipids, and they are present in two layers, bilayer. Now see these points very carefully. See here, so this is how we indicate a protein, a bilipid layer of fat, right? This is a bilipid layer, right? So these are majorly phospholipids. Second, it says the polar head towards the outer side. So this is the polar head, right? And because head is on the outer side and outer side has water, this polar head is hydrophilic. It is hydrophilic. Is that clear? Polar head towards the outer side and hydrophobic tail towards the, this is tail. This is hydrophobic. And non-polar too. Is that hydrophobic tail? The non-polar, and it is non-polar as well. So hydrophobic and non-polar tail, hydrophilic and polar tail. Is that clear? So protected. The non-polar tail of saturated hydrocarbons is protected from the aqueous environment, and that is why we call them hydrophobic. Clear? In addition to phospholipids, it also has cholesterol. So one is phospholipids and second is cholesterol. So cholesterol basically provides stability to the structure. Is that clear? Now, later on it was found, see, the ratio of protein and lipid will vary from every cell to cell. For example, in case of erythrocyte, it has 52% protein and 40% lipid. Is that clear? This ratio will vary, but in case of RBC, 52% protein and FR, can you remember the sound? P for protein, P for per, no logic, but just remember 52% proteins and 40% lipids. Clear? Now, other than lipids, we know there are proteins present. Now we can classify proteins on the basis of ease of extraction. See carefully. If the proteins are on the outer side, so those proteins can be easily extracted, we'll call them peripheral or extrinsic proteins. While those which are within this membrane, so integrated with this membrane, they are difficult to extract, so we call them integral or intrinsic proteins. Are these clear? Peripheral on the outer side, integral partially or wholly buried in the membrane. So this is the structure. Now the best structure that we follow till now, that is given by Singer and Nicholson in 1972. This is known as fluid mosaic model. What does this say? Quasi-fluid nature of lipid. Very important. Quasi-fluid means not totally fluid. If the plasma membrane would be totally fluid, we won't have a structure at all, right? Quasi-fluid means it can move, but there is limitation to it as well. So enables lateral movement of proteins within the overall layer. Is it clear? And this ability to move within the membrane is known as fluidity. Now, what is the importance of this quasi-fluid nature? It helps in cell growth, formation of intercellular junctions, secretion, endocytosis, as well as cell division. We already know plasma membrane is selectively permeable. Now, that means not everything can come and means it can enter or leave. It can come and go, right? So, There are different types of methods of transport for things which are allowed within the cell. One is active transport, second is passive transport. Active transport is always by using ATP. For example, for ions like sodium and potassium pump, we usually use the term pump for active transport only. Then passive transport, no ATP used, it will move from higher to lower concentration. So in passive transport, we have three different techniques. Simple diffusion for neutral solutes. Water moves through osmosis, and then there is a special movement called facilitated diffusion. Facilitated diffusion occurs by for polar molecules which require a carrier protein. Is that clear? Neutral through simple diffusion, water through osmosis, and polar by the help of carrier proteins. So because they are using a facility of a carrier protein, we call it facilitated diffusion. Are we clear? Please tell me, are we clear? So polar molecules cannot cross through the non-polar lipid layer, so they require a carrier protein. So they are moving from higher to lower, hence it is passive, no ATP will be used, but they need a carrier molecule to take them from one layer to another. Is that clear? So it is known as facilitated diffusion. Please tell me these points are clear to all. Let's talk about cell wall. It is non-living. It is rigid, gives shape to the cell, protects cell from the mechanical damage and infection. Cell to cell interaction provides a barrier and also prevents it from bursting. We have read that, right? Algal cell wall made up of cellulose, galactans, mannans, and minerals like calcium carbonate. While plant cell wall made up of cellulose, hemicellulose, pectins, and proteins. Is it clear? Primary wall is made up of, primary wall is capable of growth, but this diminishes as the cell matures. And the secondary wall is formed inner to the side of the cell. Is that clear? Now, middle lamella is common to two cells. So, between the two primary walls, we have middle lamella, and it is made up of calcium pectate. See, cell wall and middle lamella can be traversed by a cell junction, plasmodesmata. Plasmodesmata is a cell junction only found in plant cells, not in animal cells. Then talking about endomembrane system, very frequently occurring question, PYQ. So what is the composition of endomembrane system? Endomembrane system consists of four components: ER, GB, lysosomes, and vacuoles. The function of these four organelles is well connected, interconnected. That is why they form endomembrane system. And it's written mitochondria and chloroplast, and they are not coordinated with the above system. That is why they are not a part of it. Is it clear? Talking about endoplasmic reticulum, see, most important things. ER divides the intracellular space into two distinct luminal. Luminal means the space bounded by these structures, and outside everything is extraluminal. So extraluminal is basically cytoplasmic. Then two types of RER. Those with ribosomes on the surface, they will appear rough, so RER. And without ribosomes, they'll appear smooth, so we call them SER. RER involved in protein synthesis, and they're continuous with the outer membrane of nucleus. You have to learn these points very carefully. While SER has no connection with the nuclear membrane, not joined, not fused with the ribosomes, so not involved in protein synthesis. Rather, they are a site for synthesis of lipids and some steroidal hormones. Steroids are basically lipids. Is this point clear? Then talking about Camilo-Golgi. Camilo-Golgi. See here. One, the Golgi cisternate consists of cisternae and vesicles. Cisternae will have two phases: convex, cis and forming phase, one thing, and concave, transverse and the maturing phase. Is that clear? You have to learn these words. Concave is trans and also the maturing phase. Is that clear? So basically, the immature protein will fuse with the cis phase, and while it passes through different cisternae, it gets matured, and the final mature protein will exit through which phase? Trans phase. Is that clear? Cis and trans are entirely different but interconnected. Now, Golgi apparatus functions. Look at this. First function is packaging of materials. Second, intracellular targets are secreted. ER fuse with the cis phase, just told you. ER vesicles will fuse with the cis phase of Golgi apparatus and move towards the maturing phase. Clear? Now, very important, Golgi apparatus is important site for the formation of glycoproteins and glycolipids. See, protein and lipid by RER and SER respectively, but glycoprotein and glycolipid formation is by GB. Are we clear till here? Talking about lysosomes, guys, they are membrane bound vesicular structures for the process of packaging, right? And very rich in almost hydrolytic enzymes. So like this hydrolases, lipases, proteases, they're active at pH, at acidic pH. Talking about vacuole, bound by a single membrane called tonoplast, can occupy up to 9% of the volume. Tonoplast facilitates the transport of number of ions. Very important line. See, vacuole is like a storehouse, right? So as compared to kitchen, you have more things stored in higher concentration in your storehouse, right? And you bring out things as per the need. You don't keep the whole bulk in your kitchen, right? Similarly, the bulk is within the vacuole. So you keep on putting things, you keep on storing things in your storehouse, in your vacuole, even though they are already in plenty. So tonoplast will facilitate the transport of number of ions or other materials against the concentration gradient. Why? Because it is a storehouse, right? Now, in amoeba, there is a special vacuole called contractile vacuole for osmoregulation and excretion. And in many cells like protista, food vacuole is formed by engulfing. Amoeba is also a protista. In paramecium also it will be formed. Let's talk about mitochondria. So, mitochondria is the powerhouse of the cell, double membrane structure. It will have its own prokaryotic DNA and prokaryotic ribosomes, so semi-autonomous also. See here, the inner compartment filled with dense homogeneous substance called matrix, right? Okay, the two membranes have their own specific enzymes associated with the mitochondrial function, and it is a site for aerobic respiration. So powerhouse, single circular DNA, few RNA molecules, ribosome 70S type, components required for the synthesis of proteins, so semi-autonomous, and it has prokaryotic origin, hence it divides by fission also. Is that clear? Then talking about plastids. Plastids are found only in plant cells, all in plant cells or in euglenoids. We have read about it, right? Now see, depending upon the type of pigment present, we have different types of plastid: chloroplast, chromoplast, and leucoplast. Now, chloroplast contains chlorophyll. Chromoplast contains fat-soluble carotenoid pigments like carotene and xanthophylls, all those red, yellow pigments, right? Leucoplast doesn't have a pigment. Instead, it is colorless and stores nutrients. Now, based on the type of nutrient stored, leucoplast can be amyloplast. Amyloplast will store carbohydrate. Amylo word is always related to sugar. Is that clear? Carbohydrates, usually starch. It stores starch, like in potato, rice, right? Then aleoplast. Aleo word is for oil. It stores oil and fats. So all the oil seeds, they have lots of aleoplast. Whereas aleuroplast stored protein. Do you remember aleuron layer? It was a protein layer. So aleuroplast store proteins. Is that clear to all? Now see, the number of chloroplast will vary. It is found maximum in mesophyll cells of leaves because that is the main site of photosynthesis. The shape of chloroplast will vary. Number will also vary. For example, see, Chlamydomonas has one chloroplast. Do you remember that cup-shaped chloroplast in Chlamydomonas? Yes. And green algae chloroplast. While 20 to 40 chloroplast per cell in mesophyll. Now, like mitochondria, there are some similarities between mitochondria and chloroplast. Both have two envelopes. Both have their own 70S ribosome. Both have their own single circular DNA, prokaryotic origin, right? Inner chloroplast, see, is relatively less permeable. Important line. Inner one is less permeable. Now, we know that there are flattened membranous structures, thylakoid stacks known as grana, intergranal thylakoids, stromal lamellae, we have read all these. right? And this is also what I've told you. Talking about ribosomes, granular structures first observed by George Pallet. So we also call them Pallet particles, made up of RNA and proteins. So this is something we have read in genetics. Not surrounded by any membrane. Already told you there are two membrane-less structures. One is ribosome, second is centrosome. So 80S and 70S both are present. 80S are present in the cytoplasm or attached to the RER, while 70S are found within mitochondria or chloroplast. So see their subunits, 80, 60 plus 40, this happens only in biology. So 60S and 40S will make 80S, and 50S and 30S will make 70S, and only a biology student can understand. So maths is failing here, right? Now see, what does S stand? Actually stand for Swedberg's unit or sedimentation unit. So it is a measure of, indirect measure of density and size, not direct measure. Clear? And this actually explains why 60 and 40 makes it S80 and 30 and 50 makes it 70 and not 80. Now cytoskeleton coming here. So cytoskeleton, network of filamentous proteinaceous structure consisting of microtubules, microfilaments and intermediate filaments, right? And what is the function of cytoskeleton? Mechanical support, motility, maintenance of the shape of the cell. Now in cytoskeleton, guys, these components of cytoskeleton will form many other structures. See here, cilia, flagella and so on. So both cilia and flagella, they're covered with the plasma membrane, but the core is known as exoneme. Exoneme has 9 doublets, radially arranged peripheral microtubules. What is the difference between tubule and filament? Tubule is hollow, cylindrical structure. Filament is solid, right? Thread-like structure. Now see, so in exoneme, what do we have? We have 9 doublets of microtubules and a pair of centrally located microtubule like this. These are peripheral microtubules, doublets of it, and centrally placed 2. Is that clear? Connected by bridges enclosed by the central sheath. Peripheral ones are also connected to the central ones by radial spokes, and all the outer peripherals ones are interconnected by linkers. Cilia and flagella emerge from centriole-like structure known as basal body. Is that clear? Now you see centriole, basal body, they will have similar structure. This cilia and flagella will have 9 plus 2 arrangement. 9 on the outer side, 2 in the center, right? 9 on the outer side are doublets and 2 in the center, right? But when we talk about centrioles, they will have 9 plus 0 arrangement. Why 9 plus 0? But because there will be no microtubule or filament in the center. It will be a protein hub. And also the 9 on the periphery will not be doublets now. They will be triplets. See here, containing 2 cylindrical structures called centrioles forms a centrosome, and there is amorphous pericentriolar material. Both are lie perpendicular and have a cartwheel-like structure. How is the cartwheel-like? There is one central hole, and then there is a periphery, there is a rim, right? So that cartwheel-like structure. So 9 evenly placed peripheral fibrils of tubulin protein, and now they are not doublets, there are triplets here. Central region is made up of a proteinaceous hub. All the triplets are also connected to the hub through the radial spokes and to each other through linkers. Same. Is that clear? The centrioles form the basal body of cilia and the spindle fibers and give rise to spindle apparatus. Talking about nucleus, guys, discovered by Robert Brown. The material contained is stained by basic dyes, given the name chromatin by Fleming. Now, if you want to study the structure of nucleus, the best stage is interphase. Why? Because while it is preparing for division in the interphase, the membrane is intact, right? So membrane is intact, so the boundary is intact. It is the best stage. Interphase stage. And what does interphase nucleus looks like? It has nucleoplasm, it has nucleolus, nuclear pores, the nuclear membrane, and of course the genetic material. Now these nucleoli, one or more spherical bodies, okay, and it is made up of rRNA. It is the rRNA synthesis, site of synthesis. Is that clear? So if a cell is involved in protein synthesis, it will have more number of nucleolus or nucleoli. Is that clear? Then nuclear envelope is also double layered, and where it fuses, it forms the nuclear pore. Clear? Then it runs parallel to each other within a space known as perinuclear space, which forms a barrier between the materials. Are these points clear? See, pores are formed by the fusion of the two membranes. Pores are the passage through which movement of RNA and protein molecules will take place in both the directions. Now, mature cells even lack nucleus. So, you should know the names, examples of the cells which lack nucleus. One is RBCs in mammals, most of the mammals, and sieve tubes in vascular plants. Is this clear? That is why they need companion cells. So, nucleolus active rRNA synthesis site, we have discussed that. Chromatin material contains DNA and histones as well as non-histones to form the chromosome. Now there is the site of primary constriction known as centromere, and around the centromere we have two disc-shaped structures known as kinetochore, and around this disc-shaped structure the spindle fibers will attach. So kinetochore is a site for spindle attachment. Is that clear? Now centromere holds the two chromatids together. So there are different types of chromosomes based on the position of centromere. So you will always know, you should always know the basis. So what is the basis? One is, so the basis is position of centromere. So according to this position of centromere, we have four types of chromosomes. See here, we have four types of chromosomes. First is metacentric. Centromere right in the center, the upper and the lower, P arm and the Q arm, equal size. See, equal arms. Metacentric, centromere slightly away from the center. So P arm is slightly shorter or Q arm is slightly longer, or the vice versa. Then acrocentric, A for acro and A for away. Centromere quite away from the center, close to the end. So one is extremely short while other is extremely long. Is that clear? And then we have telocentric, where we have a where we have centromere towards the tip. Is this point clear? You should be able to identify the diagrams and also identify the exact definition. And then guys, other than primary constriction, we might have a secondary constriction also. So the secondary, beyond the secondary constriction, the part of chromosome is known as a satellite. So gives the appearance of a small fragment rotating around the main body, that is satellite. That's why we call it. It is non-staining secondary constriction at a constant location. So please let me know that all these points are clear. Is that clear to all? Yes guys, is this point clear to all? So guys, the name of my Telegram channel is Tina Ma'am Biology, and you will find all these PDFs in the Telegram channel. Is that clear? All right. Clear? All right, guys. Now let's do one thing. Let's quickly complete the next chapter and we'll stop it there. Let's complete one more chapter. How many of you are still up for another chapter? One more chapter. Can we complete cell division here? Let me know. Please give me a thumbs up if you are up for another chapter. Okay? One more chapter, shorter chapter. We'll underline all the important lines, right? And then we'll keep rest for tomorrow then. Don't worry. I'll not interfere with your chemistry or physics class, sorry. All right. Let's do one more chapter, guys. We'll be able to complete 10 chapters today. Sorry, 9 chapters today. Don't worry. We'll complete rest of them tomorrow. I'll take another session. All right. Just a second, just a second. All right, guys. Let's do one more chapter. Not biomolecules. Let's do cell cycle. Yes, please. All right. First line. The sequence of events by which the cell duplicates its genome, the synthesis, and eventually divides into two cells. This whole sequence is known as cell cycle, where there will be cytoplasmic increase, and that cell growth is a continuous process. Very important line. DNA synthesis occurs only during a specific stage, and you know that is the S phase. Is that clear? Now, moving on to the phases. So, typical eukaryotic cell divides every 24 hours. A yeast cell will divide only about in 90 minutes. So every cell cycle consists of two phases, interphase and the M phase. So see, M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, that is karyokinesis, and division of cytoplasm is known as cytokinesis. Is that clear? Cell growth, DNA replication occurs in an orderly manner, and you know cell growth will keep on continuing. It is a continuous process. Interphase is divided into three phases, G1, S and G2. Is that clear? Now, during the S phase, the cell is metabolically active and grows continuously but does not replicate the DNA. In S phase, DNA synthesis will occur, DNA amount will double up, but the number of chromosome will remain same, no increase in the chromosome number. So these are the points to be remembered. I am discussing questions around this topic in my special classes, guys. So don't forget to watch those classes and attend those classes, practice those classes, those questions in the special classes. We are discussing tricks how to solve the questions in the quick manner. What are the points to be taken care of? So make sure that you are attending all the special classes. My question practice is actually only in special classes. I'm not taking any question classes here. Is that clear? And question classes are important. Practice sessions are important. Yes. Now, let's move on. S phase, DNA replication, centriole duplication, and even histone formation occurs during S phase. Protein synthesis occurs during the G2 phase while cell growth continues. So straightforward questions match the following: DNA replication in which phase, protein synthesis in which phase. See, there are different types of protein synthesis, but if they simply say protein synthesis, you have to match it with G2 phase. Is that clear? Now, moving on to next, cells that do not divide exit the G1 phase and enter inactive stage called quiescent stage or G0 stage. But remember, the cells at this stage remain metabolically active, no longer proliferate, do not divide, but they remain metabolically active, and it is during this phase that they differentiate. They become functionally active. Is that clear? Now, M phase, the most dramatic period. Now, prophase, see, it is in the S phase and G2 phase that the new DNA molecules are not distinct, right? Although they are multiplying, DNA content is doubling up, but they've not condensed to form the chromosome. So that composition occurs during the prophase. The chromatin material, genetic material will condense to form chromosomes here. So chromosomal material condenses. Centrosomes undergone duplication during the S phase. They will move towards the poles, radiate out the microtubules called the asters, and two asters with spindle fibers form the mitotic apparatus. So see the composition of mitotic apparatus. Two asters and with their spindle fibers, this will form the mitotic apparatus. Is that clear? Now, at the end of prophase, guys, all these important structures like GB, GB, ER, nucleolus, nuclear envelope, they are all disappeared. They will come back again in telophase, but in prophase, by the end of prophase, they are all gone. Now see, complete disintegration of nuclear envelope marks the start of the second phase. So as soon as prophase is vanished, it is the beginning of metaphase. Second characteristic of metaphase is that all the chromosomes, they come to lie on a equatorial plate, right? Although it is an imaginary line, just like we talk about equator with respect to Earth, equatorial plate is also imaginary, but they come to lie in the central area. That central area, that is called equatorial plate or metaphasic plate. See, now chromosomes have condensed to their maximum already. So if you want to study the structure of chromosome, metaphase is the best stage, right? Chromosome made up of two sister chromatids held together by centromere, small disc-shaped structures called kinetochores. They are the site for attachment of spindle fibers, already told you, right? Characterized by all the chromosomes coming to lie at center, one chromatid of each chromosome connected by the kinetochore to the spindle fiber, and the sister chromatid connected to the kinetochore on the opposite side. Is it clear? See, metaphase stage, attached to kinetochore and spindle equator such that they are aligned. Then talking about anaphase. So this is a metaphase. Anaphase, metaphasic plate. Now chromosomes which are arranged on the metaphasic plate, they will split in such a way that sister chromatids will separate. See here, what is the characteristic line here? Sister chromatids split and centromeres will split and sister chromatids will separate and move towards the opposite poles, right? In such a way that centromere will be towards the centriole and the chromatids will trail behind. Yes, telophase, just the opposite of prophase. Chromosomes, now they have reached to the poles, they will start undergoing decondensation. So from chromosome, it will come back in the chromatin fiber stage, right? So they will lose their identity. Nuclear envelope will redevelop. Nucleolus, GB and ER will reform. So you can get these questions directly as statement based or match the following. So be ready for all type of questions. And after karyokinesis has occurred, cytokinesis will follow. This cytokinesis can occur by two methods. In animal cell, it will appear by the appearance of furrow formation. Is that clear? It is furrow formation. So furrow keeps on deepening on the inner side, and that furrow will separate the two daughter cells, right? While in plant cells, there is cell wall formation. Now, cell wall formation begins with the formation of simple precursor called the cell plate. And the cell plate, it represents the middle lamella. And how does cell plate cell plate formation begins? By the aggregation of GB vesicles in the center. Golgi body vesicles. Is that clear? Now, in some organisms, karyokinesis is not followed immediately by cytokinesis. Now, this leads to multinucleated condition known as syncytium. For example, liquid endosperm in coconut. Is that clear? Now, what is the significance of mitosis? Some lower plants and some social insects like even bee, honeybee, haploid cells divide by mitosis. Now, production of diploid daughter cells with identical genetic complement. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. So, it is essential for the cell to divide to restore the nucleocytoplasmic ratio. Very important. So, what is the reason behind cell division? What is the stimulus behind cell division? This is the stimulus. Disturbance of nucleocytoplasmic ratio. One of the stimuli, of course. Is that clear? So all this repair and healing and continuous growth, and in some cases the formation of cells like gametes also occurs by mitosis. Is that clear? Then talking about meiosis, majorly seen in gamete formation in diploid organisms, right? So it occurs in two sequential cycles, meiosis 1 and meiosis 2, but single cycle of DNA replication. So interphase is one only. Is that clear? Then meiosis involves pairing of homologous chromosomes. Now this did not occur in mitosis. Clear? 4 haploid cells are formed at the end of meiosis 2. So meiosis 1 and meiosis 2. Now out of this meiosis 1, prophase 1 is the most elaborate step. So occurs in 5 stages. See, lazy, zebras, ponderoid, disasters, leptotene, zygotene, pachytene, diplotene and dikinesis. Let's see the quick points about leptotene. Leptotene can be easily observed under light microscope also. You start seeing the compaction, condensation of chromosomes and then it is followed by the second stage called zygotene. In zygotene, the homologous chromosomes start coming together. Pairing up of homologous chromosomes occur. In order to keep the homologous chromosomes together, you need synaptonemal complex. So that is also formed here. Now, the pairing up of homologous chromosomes is known as synapsis. What brings them together is the synaptonemal complex. So when two homologous chromosomes are lying side by side, that stage is called bivalent. And bivalent have how many chromatids? 4 chromatids, 2 of each. So that is also known as tetrad. Is that clear? Now the first two stages, that is leptotene and zygotene, are relatively short-lived. Followed by the third stage, that is pachytene, and one very important event is going to occur here in prophase 1. In pachytene, that means in pachytene, crossing over will take place. So genetic exchange, the genetic material will exchange between the crossing over between the two non-sister chromatids of homologous chromosomes. So read the statement carefully, right? And now so what will happen? New combinations will form. So recombination is occurring. So during crossing over, recombination nodule is formed, and crossing over is an enzyme mediated process involving the enzyme recombinase. Please tell me these points are clear, right? They're completed by the end of pachytene, and then once the crossing over is done, there is dissolution of the synaptonemal complex, and this starts in diplotene. Dissolution in diplotene. What will happen? Homologous chromosomes will separate from each other except at the site of crossover. And now this site of crossover, this X-shaped structure, this is known as chiasmata. Now, as a matter of fact, in some oocytes, in some vertebrates, in oocytes, diplotene can last for months or years. Like in our case, it lasts for 10 to 11 years till we reach the puberty. Our primary oocytes, they are lying in this diplotene stage, right? Now, final stage is the dikinesis, where terminalization of chiasmata take place. Now, this paragraph is the most important paragraph. We get so many questions out of this, right? And we are practicing those questions in special classes. Now, metaphase, bivalent chromosomes align on the equatorial plate, just like in mitosis, but the difference is here there will be 2 equatorial plates formed, such that on each equatorial plate, one of the homologous chromosomes will align. See, these are homologous. So one of the homologous is on one equatorial plate, other on the other equatorial plate. Are you getting that? Now, metaphase 1 is followed by anaphase 1. Now, in anaphase 1, there will be no splitting of the centromere. Sister chromatids will not separate. Homologous chromosomes will separate. So the whole chromosome will move there on the other pole. Are we getting that? Sister chromatids remain associated here. While in telophase, just the opposite, all the nuclear membrane, nucleoli and ER, GB, everything will come back. Is that clear? That will be telophase 1. So 2 daughter cells will be formed, 2 daughter nuclei will be formed, and now this will be followed by cytokinesis. It may or may not be. Cytokinesis may occur at the end of meiosis 2 together, or it may occur here and then each cell will individually undergo meiosis 2. Is that clear? Now there is this short break between the meiosis 1 and meiosis 2, and this is known as interkinesis. Now during interkinesis there is no replication of DNA. Remember that. Now meiosis 2 is just like mitosis. So consider that each daughter cell of meiosis 1 is undergoing mitosis. Same to same. See, in anaphase 2, there is simultaneous splitting of centromere. This is similar to anaphase of mitosis. Everything. Is that clear? So guys, this was meiosis 2. So at the end of telophase, we have 4 daughter cells formed. That is always the case. In meiosis, what is the significance? Conservation of specific chromosome number of each species. So why after generation and generation after generation, the number of chromosome remains same? It's because of meiosis, right? And then it increases the genetic variability during crossing over, right? So it is very important step for the process of evolution. Is that clear to all? So guys, we will stop here. Yes, we'll stop here. And I'll continue the session tomorrow. Let's meet again at 11 or 12 and we'll continue rest of the chapters tomorrow. Is that clear? And probably I'll take another class tomorrow itself from 7 where we will be studying the class 12 biology as well. Is that clear? So I'll see you tomorrow. Don't forget to join me. And tonight at 9 p.m. we have a special class. Bye guys.